Hi everyone, welcome to our mini student psychiatry conference. My name's Dee and I'm the president of Sheffield PsychSoc, and co-hosting with me tonight are Benjamin Gears, president of Manchester PsychSoc, and Reese Hill, vice president of Newcastle PsychSoc. Just to let you know, this conference is being recorded. This conference aims to introduce you to the fascinating world of psychoneuroimmunology, which incorporates a wide range of specialties, not only psychiatry, immunology, and neuroscience, but also psychology, pharmacology, molecular biology, infectious diseases, the list goes on. We have three incredible speakers tonight. We hope you enjoy yourselves and further increase your knowledge on the inextricable link between the mind and the body.
Yeah, hi guys. My name is Reese and I'll be representing Newcastle Psychiatry Society. Just to quickly run through just a few things. As you can see, the messages coming up here. All of you are muted, but if you'd like to ask any questions at any point, if you put those into the chat, and after each talk, there's a Q&A session lasting for approximately 15 minutes, during which time you could either read out your question or we'll do it on your behalf. If at any point there's some sort of tech difficulties, again, put those in the chat and we have a few people on hand to kind of help us out with that sort of thing. Thank you.
Hi everyone, my name is Benjamin and I'm the president of Manchester Psychiatry Society. We would just like to start by saying a huge thank you to our speakers tonight. The talk's looking incredible, and the speakers put a lot of effort into not only pre-recording the talks, but also being here tonight to answer your questions. So we're incredibly grateful for the hard work that's been put into this. Our first talk, which is going to start now, is by Professor Peter Jones, and it is titled Gene-Environment Interactions: How Genes and the Environment Shape Our Mental Health. Peter Jones is Professor of Psychiatry at the University of Cambridge and an honorary consultant psychiatrist at the Cambridgeshire and Peterborough NHS Foundation Trust. Professor Peter Jones leads the Cambridge Epicenter Group, which works at the interface of neuroscience, population-based research, and clinical psychiatry to delineate epidemiological, genetic, neurobiological, and psychological factors that influence the cause and outcomes of mental illnesses. Professor Jones's talk will outline and summarize key concepts and principles of gene-environment interactions within psychiatry, demonstrating how neurobiological and social factors can interact to cause psychiatric disorders. So without further ado, here's our first talk.
Good evening, everyone. Thank you to Benjamin Gears and colleagues for inviting me to talk to you this evening about gene-environment interactions in psychiatry. I'm going to go over some definitions with you about gene-environment correlation and interaction. Then I'm going to think about the complexity of the ideas for psychiatry, because environments, genes, and phenotypes are complex for us, and introduce the idea of an endophenotype, something closer to genes than final diagnostic categories, also called intermediate phenotypes sometimes. Then I'm going to go through some classic examples of gene-environment interactions for antisocial behaviour, depression, and drug effects, and draw out some of the methodological caveats in this field. But I'm not actually going to cover genetic epidemiology and statistical methods, although I've put some further reading at the end for those of you who want to pursue that. And then think briefly where we might be going in terms of gene-environment interactions in psychiatry in the light of the examples I give you. So first of all, some definitions. So gene-environment correlation. This is where genetic variants influence environmental exposure through genetic effects on behaviour. So it's different from gene-environment interaction. And there are different ways to think about gene-environment correlation, and generally thought about in three groups. They sometimes go by different names. So passive gene-environment correlation is where a genetic effect on a trait makes that trait look causal. It's a sort of special case of confounding. So an example of this might be where someone with an antisocial personality disorder abuses their child, and the child has a poor outcome, perhaps with antisocial personality themselves, and it might look as though the abnormal child rearing has led to the child's psychiatric outcome, whereas actually it could be a more direct genetic effect. Similar examples with the DRD2 dopamine receptor polymorphisms for ADHD, where a mother might transmit that genetic effect to the child that causes their ADHD, but in the mother that might lead to abnormal behaviour that looks as though it might be causing the child's poor psychiatric outcome. Then there's what gets called evocative gene-environment correlations. So it might be that a genetic behavioural trait or a disorder is eliciting a certain sort of environment. And a good example of that is living with someone with a severe mental illness might cause tensions in the family. They're not causing the individual psychiatric disorder, they're being elicited by them. So there might be a correlation between genetic effects and the environment for that person. And thirdly, there's more active gene-environment correlation, and this is where a genetically influenced trait leads to someone seeking out an example. So gambling, pathological gambling, for instance, has some heritability, and you might find more of those people in Las Vegas, and it's their genetic predisposition to gambling that's leading them to be there, not the other way around. So gene-environment correlation to one side then. What we're talking about this evening is gene-environment interactions, or G times E. And this occurs where two different genotypes respond to environmental variation in different ways. So genetic alternatives have different responses to an environment. Now, I was reminded when I was invited to give this talk of something way back in my career when I was a medical student. And this was in my second year exams. And it was in genetics, and we'd been prepped for a certain exam question. And we knew we were going to be asked to write about an example of gene-environment interaction. And I decided to write about something a bit different, the lac or lactose operon in E. coli. And in bacteria, RNA is expressed in what are called operons, which are little collections of proteins which serve biological processes. And E. coli preferentially metabolizes glucose, but if it's in an environment where there's little glucose and alternatives like lactose, then it can switch on the transcription of genes that will lead to lactose being an appropriate food. So it transcribes lactase, cleaving the lactose into galactose and glucose. And this rather lovely figure of Nobel Prize winning work shows that in the top we've got the system is repressed, and there's the repressor sitting on the RNA, and the RNA polymerase is unable to transcribe the operon. And in the presence of lactose, the repressor gets inactivated, and the polymerase can translate the operon. And the E. coli can feed off the glucose that's produced. All very good. I thought that was gene-environment interaction. But unfortunately, that was the wrong answer. And I almost failed the paper. I had to have a special viva. Unfortunately, I was able to answer. And I'd repressed that thing from my student days, but it came back to me preparing for this talk. And of course, actually, I was wrong, because that wasn't an example of genetic variants leading to something different in environmental variation. It was a single genetic phenomenon. What, of course, the examiners were looking for was classic examples of gene-environment interaction, like sickle cell anemia, autosomal recessive. You'll all know all this, where heterozygotes show some resistance to malaria, hence the persistence in the population. Another one would be G6PD deficiency, might lead to hemolytic anemia in the presence of certain environments, fava beans, drugs, but again, can protect against malaria. Phenylketonuria, another autosomal recessive example. In the homozygous situation, catastrophically raised phenylalanine levels can lead to severe CNS effects, learning disability, seizures, mental health disorders. You treat it by manipulating the environment, keep phenylalanine low. That persists in the population because heterozygotes with slightly raised phenylalanine women show some resistance to a fungal toxin that may lead to spontaneous abortion, hence the preservation in the population. So I knew that's what I should have written about, and that's what came up in the Viva. But of course, those examples, PKU, that does have some mental health sequelae, but in general, they're about simple environments and simple genes. Now, it's more complicated than that in all of medicine, but particularly for psychiatry. So one complication is these ideas of clinical and causal heterogeneity. So single causes can lead to multiple outcomes. In psychiatry, early life adversity, child abuse is associated with a wide range of adverse psychiatric outcomes. You can also have causal heterogeneity, so you can have a single outcome that has multiple causal contributions, and that's the case for many psychiatric disorders. And in psychiatry, our environments are very complex, and we have multiple genes, as we know from genome-wide association studies, use of polygenic risk scores, etc. So before we can think about gene-environment interaction in psychiatry, we've got to think more carefully about what the environment is for the brain and for the mind. And there are several environments that the brain and mind interact with. So some of them are relatively simple, harking back to those chemical examples of G times E I was talking about earlier. So there's an external physical environment that the brain and mind live within. There's an internal microenvironment, the internal milieu. The presynaptic environment might be key for CNS function and for mental health. Then we've got where it begins to get a bit even more different for psychiatry, because other organs don't create their own environment in the same way. Our brain and mind has to represent our external environment through perception, emotional reactions to those perceptions. We're making different decisions and choices. So we're creating our environment. And it's also linked to the way we represent ourselves within the environment. So, for instance, if we were live this evening and I made a little joke and I heard you laugh, that will be a change in my environment. And if I thought you were laughing at my joke, then I'd be very pleased. I'd get a little squirt of phasic dopamine in my striatum and I'd be satisfied with life. However, if I heard you laughing, the same external stimulus, physical stimulus on my ears, interpreted as laughter, and I realized that you were actually laughing at me because I had food down my front. I hadn't tidied up after I'd spilt my lunch everywhere. Then that same environmental stimulus would make me embarrassed. I wouldn't get a dopaminergic reward. I'd get high cortisol, stress hormones that could be neurotoxic. So the same environment, it's my meaning of that environmental exposure that's leading to very different internal reactions. So the environment is complex in psychiatry. And in fact, for some periods in the relatively recent decades, that's all been ignored. So schizophrenia, for instance, 20 years ago, we'd be thinking that schizophrenia is a largely genetic disorder. But we know that's not the case. So I've been involved in this study, the European Union Gene Environment Interaction Study, EUGI, a large multi-center study set up to look at gene environment interactions in psychotic disorders. 17 settings in 6 countries. And looking very simply, that study, here's Hannah Youngsma here, who was my student at the time, did this analysis showing in the different sites which are on these different, in this column here, Spain, Italy, Spain, Cambridgeshire and Peterborough, where I come from, France, Italy, etc. The incidence of psychotic disorders per 100,000 person years is very different. So these are different estimations of the incidence rate, crude and then adjusted for different socioeconomic variables. And you can see that these 3 sites here in Paris, in a very poor area around the périphérique in Paris, in Amsterdam and in southeast London, the incidence per 100,000 person years is much higher. So hinting at some environmental factors that we're going to look at gene environment interaction to see if they can be explained. So a complex environment is important in that disorder. And along with James Kirkbride and others at UCL and Cambridge, we've actually taken that environmental variation to try and use it as a predictor variable for service planning using a tool called Synaptic. And here you see a map of England with small areas coded for the numbers of new cases of psychosis, with red as high numbers of cases in the next year, green down as low. And you can see if you look across the map of England, then there is heterogeneity arising out of the environment. We don't think that there are genetic variations that can be leading to that sort of spatial distribution. So the environment's important and it's complex. In summary, the key environmental factors that we think of in psychiatric disorders, particularly exemplified by first episode psychosis, are migration, urbanicity, early life obstetric events, childhood adversity, and more recent life events occurring proximal to a psychiatric disorder. Those are the rate ratios or odds ratios of association. And I'm going to take you through later some gene environment interactions for the last 3 of these. And I'm giving you some further reading about urbanicity as an environmental factor. And actually it's more of a gene environment correlation, with genes might be leading. There's a small genetic contribution to where people live, believe it or not, but I'm pointing to that in further reading for you. So here are some environmental factors that I'll pick up later. What about genes? Well, when I was a medical student, we knew that psychiatric disorders were highly heritable. Family studies, pedigree studies, adoption studies were the bedrock of early days of genetic epidemiology, and more recently, of course, we've got more molecular-based methods. Many of you will be familiar with this Manhattan plot from the 2014 psychiatric genomics collaborative study that was published in Nature, which showed 128 independent associations across 108 different genes. And many of those were candidate genes in the brain, particularly synaptic genes, to do with dopaminergic transmission, glutamatergic transmission. But many of the biggest hits were actually in unexpected areas, including the immune system. And I know you've got talks later about psychiatry and immunity, so I wouldn't say much more about that. So the genetics of this disorder, we now have genome-wide association studies for other disorders, autism, depression, OCD. It's complex. These are polygenetic disorders. And we also know that there's some of that heterogeneity that I was talking about earlier, causal heterogeneity. So there are genetic correlations across psychiatric phenotypes. This figure comes from the Brainstorm Consortium, showing correlations coded as very high through to low between, if you look at major depressive disorder here, a lot of genetic overlap with ADHD, major overlap with anxiety disorders, schizophrenia and bipolar disorder, a lot of genetic architecture overlap between those disorders. So again, genetics is not simple, and we've got to take that into account when we think about gene-environment interactions. Now, going back a couple of decades, before we knew about any of the complexity of genetics, certainly, we've begun to get some theoretical models that could accommodate the idea of gene-environment interaction. And I think the first and most important of these was the so-called stress vulnerability model put forward by Zubin and Spring. And here, vulnerability along the bottom axis here could be anything, but it's often thought of as diathesis or genetic vulnerability. And stressful events in this model is plotted on the y-axis. And you see there's an interface here, a function between stress and vulnerability, across which you can move from mental health to mental ill health or mental disorder. So there's a stress threshold. And the higher your vulnerability, one example of that vulnerability would be genetic diathesis, the lower the stressor needs to be to trigger psychiatric disorder. So there's a function here between genetic risk and environmental stress induction, potential interaction. Now, this has been taken a little bit further in recent years with this idea of an endophenotype, characterized, as I've said here, by simpler neurobiological and genetic antecedents than psychiatric disorders. So genes are a long way from the sort of behavioral, emotional constructs that we call diagnoses. So what's happening in between? Well, we can posit that there are endophenotypes or intermediate phenotypes. So they fill the gap between genes that code for proteins and these complex phenotypes and behaviors we call diagnoses. I'm going to show you a nice figure summarizing an example of different endophenotypes that have been thought of in systems approaches to schizophrenia. The original figure comes from a lovely review paper by Gottesman and Gould that, again, I've put in the further reading. So let's just go through this, this figure. So here you've got Zubin and Spring 30 years on. So on the y-axis here, we've got liability to schizophrenia. It could be liability to any disorder. And on this axis, we've got age. And then on this Z-axis, we've got a summary, a conceptualization of environmental effect or environmental stress. Could be a psychological stress, could be a chemical stress, doesn't really matter which. And here we've got two planes in 3 dimensions. This is the threshold over which you might develop a disorder. This example is centered around schizophrenia. And the lower threshold below is here a lower threshold for schizophrenia spectrum disorders, which perhaps it takes a lower genetic and environmental set to cross that threshold. And then you've got the 3-dimensional reaction surface, which summarizes someone's liability as they age. And you can see that if we take the schizophrenia threshold, then people will cross the threshold at high values on the environmental Z-axis during a certain period of the life course if they have a higher liability to schizophrenia. So that this figure combines environment, liability, and age as you move through the period of risk. And this is pre-GWAS, so what Gottesman and Gould did was to take the candidate genes from areas where there was linkage, parts of the genome where we knew there was something going on to do with schizophrenia. So these are chromosomal areas, and these are individual genes such as DISC1, dysregulated in schizophrenia, catecholomethyltransferase, NRG1, different genes. And at the time, it was felt that these different genes might exert their action on liability to schizophrenia through these endophenotypes. So glial cell abnormalities might be linked to NRG1 and PPP3CC. Working memory as a neurocognitive endophenotype might be linked to DISC1 and to catecholomethyltransferase. So this was a model trying to link in the idea of endophenotypes with an age, environment, genetic, interactive function. So let's look in more detail at what might happen if you take studies of a neurocognitive endophenotype. I'm going to talk about more general executive functions, actually, than working memory. And this gene, COMT, catecholomethyltransferase, which of course is responsible for the metabolism of catecholamines in the synaptic cleft. So what we've got here is a forest plot. I bet you'll be familiar with these, of the association between a functional polymorphism at one allele in the COMT gene. And this functional polymorphism involves valine and methionine. So the genotypes can be Val-Met, Val-Val, and Met-Met. And these are associated with different functions of the COMT. So if you look over time, these are in date order of publications of this association. So Mike Egan and colleagues come out in 2001 with 3 papers where they're showing in one of them certainly, a strong association between COMT genotype and executive functions in schizophrenia. And this set this field alight. We suddenly had a handle on endophenotypes. But then you can see over subsequent years, not many years, there's all sorts of attempts at replication, and it's all a bit of a mess, really. Here's the line of no effect. And these are the effects, 95% confidence limits, of course. And the summary effect is just about significant, actually, but a very tiny effect that COMT genotype is associated with executive functions in schizophrenia. However, if you take out the first three of these, the Mike Agan studies, and the key question here is, to what extent did this group have a really strong hypothesis about CRMT, or was this a finding that needed replication? So if you take out those seed studies, then the finding becomes insignificant. And those of you who want to follow up, I put a reference to Jonathan Flint and Marcus Manifo's paper. So the point of going through this with you is that there are some pretty basic statistical complexities in here about where these stories begin. And just to link with the other talks you'll get tonight, endophenotypes, of course, can be of all sorts. So they're not just about structure and neurocognition. Some of them may also be immunological. So here's another forest plot from a review looking at adverse child experiences and later inflammatory markers. So these aren't in psychiatric populations. This includes general population. And for 3 inflammatory markers, CRP, IL-6 and TNF-alpha, you can see that overall the pooled effects of an early adverse childhood experience seems to be associated with higher markers of systemic inflammation later on. And that's another intermediate phenotype that GE interactions might be working on. So bear that in mind when you hear the other talks this evening. So let's get down to some of these classic examples now I've given you the background. So 2 papers really set this field alight. The first one, 2002, Avshalom Caspi and colleagues studying, working in the Dunedin birth cohort. So it's a very valuable cohort for psychiatric epidemiology, about 1,100 people in the cohort. And they were working on the hypothesis that monoamine oxidase A genotype can moderate the influence of childhood maltreatment on neural systems implicated in antisocial behaviour. So there was a backstory to this, of a confusing literature. So they tested whether antisocial behaviour would be predicted by an interaction between MAOA genotype and an early environmental stress or maltreatment in the child. And they used the well-categorized variable number tandem repeats polymorphism in the promoter region of MAOA, which affects its expression. So different genotypes have different expressions of monoamine oxidase. MAOA is X-linked, so they can do this study only in little boys. So let's look at what they found. And this is an absolutely classic plot demonstrating gene-environment interaction or statistical interaction. So we'll come back to that. So what we've got here on the x-axis, we've got the childhood maltreatment classified as none, probable, or severe. So these are composite variables about being hit, neglected, emotional abuse, classified as none, probable, and severe in children. And then we've got on the y-axis another composite index of antisocial behaviour in later life. And this is stratified by the genotype. So either low MAOA activity or high MAOA activity. And if we take the high activity, you can see that, because that's in more people, so as a reference, if you like, more little boys, you can see that the chances of later antisocial behaviour do increase as childhood maltreatment increases. So probable, the index is higher than none, and severe, the index is higher than probable, so dose response. And then if you plot the same thing for the alternative genotype with low activity, you see this interaction. So a much steeper gradient between the probable and severe exposures. So this effect of maltreatment is moderated by genotype, and that is statistical GE interaction. Now, I've already mentioned with COMT and the endophenotype of executive function that these things need replication, and this work has been replicated actually. So here we've got a paper by Ferguson and colleagues. I use this one because this is working in another New Zealand birth cohort, slightly smaller Christchurch birth cohort. So they went in now specifically again testing this hypothesis, seeing if they could replicate Caspi et al. Again, they just were able to look in boys, and their findings, they say, added to the evidence suggesting there is stable GE interaction involving MAOA, early abuse and antisocial behaviour. They took the findings a little bit further. These are presented in a different way. So they're not presenting that different plot. They're presenting effect sizes from regression models of the GE interaction. And the effect sizes are plotted as these horizontal bars. So the longer the bar, the bigger the effect. And here are the different types of early life abuse. So significant sexual or physical abuse, significant sexual abuse alone, significant physical abuse. And these are now the outcomes, different types of antisocial outcomes. So hostility in early adulthood at the bottom there, conduct problems aged 14 to 16, official records of convictions going up, self-reported property offending, and self-reported violent offending. So there are different early life exposures and there are different outcomes, a range of outcomes. So quite a good test of this general hypothesis. And that you can see that the officially recorded convictions, ages 17 to 21, those effect sizes are smallest. And in a way, they're the hardest outcomes, but they're also a long way removed. You know, it takes another criminal justice system to reach that outcome. But the general pattern of results here replicates the notion that the effect of early life abuse on antisocial outcomes is moderated by MAOA genotype, so GE interaction. However, as I've said here, it's not all plain sailing, and findings have been debated and debated. Here's a nice review from 2018. And they've now got the luxury of 2 meta-analyses, not just replication in 2 studies, but 2 meta-analyses show that this statistical interaction is present. But what does a statistical effect really means in terms of something interacting with the molecular genome? So they raise a number of problems here. So the first is what happens in positive or nurturing environments? Does the effect reverse? The real or the causative environmental factor may be different. So we're measuring child abuse, but that's linked to all sorts of other things that might be going on in the home. It may be that the real outcome is unmeasured. So we've measured antisocial outcomes, but there may be an unmeasured phenotypic characteristic that is operating in the real GE interaction. And there may be other genes that are involved in a deeper way. So again, it's complex. So that was early life. What about adverse life experiences in adult life or closer to disorders? And again, it's Absalom Caspi and colleagues in the Dunedin birth cohort, again, writing in Science, probably the most highly cited paper in psychiatry, biological psychiatry of recent years. And they were now looking at the 5-HT transporter gene, serotonin transporter, and that has what's called an end-length polymorphism, exists in two forms, long allele or short allele, and again, they differ in the higher or lower expression of the 5-HT transporter protein. Doesn't really matter what the exact biology is. And they were looking at moderation of this genotype of the association between recent life events and depression. So here's what they found. And again, they're back to their classic GE normograms. So we've got 4 outcomes here. So number of stressful events occurring in early adult life, 0, 1, 2, 3, or 4 or more. And the 4 outcomes are self-reported depression symptoms, probability of major depression episode, probability of suicide attempt, and informant reports of depression, so sort of softer outcome. And again, you can see that the effect of stressful life events is different for the alternative genotype, so long versus short being the greatest contrast. So the effect of stressful life events on the liability of depression is moderated by 5-HT transporter genotype, classic gene-environment interaction. Now this, as you'd expect for a highly cited paper, this has also got a lot of airtime, and if you're going to read a good critique of this, it's almost 10 years old now, but Peter McGuffin and colleagues thought carefully about the replication studies and meta-analyses, and this is their conclusion, that the truth is there are just too many straws travelling in the same direction for us not to know which way the wind is blowing, which seems a rather enigmatic conclusion. But perhaps if you reflect on that and some of the problems I showed with the MAOA interaction, you can see that perhaps this is about as far as one can go. Lastly, I'm just going to think about cannabis as an example of an environmental factor that we ingest chemical. But it's actually, you could extend it to pharmacogenetics. So drugs are internal environmental factors that we take. But of course, interest in cannabis is largely driven by putative causal factor in schizophrenia and in affective disorders. Several reports that the effects of cannabis are moderated by genotype. And again, COMT and the VAL-MET functional polymorphism has been popular. And this has been shown for the outcome of psychosis, Honke et al., and also for cognitive endophenotypes. And this field has been extensively reviewed, and it does seem as though there's a particular collection of genes, so COMT, and then kinases, which moderate the acute effects on neurocognitive possible endophenotypes for psychosis, and a similar set of, slightly broader set of genes that influence the effects, mainly on neurocognition, of the chronic effects of cannabis. And of course, many of those genes are implicated in the causes of schizophrenia. So those are 3 examples: early life environment and later antisocial behaviour, adult environment and adult disorder, and cannabis, looking at MAOA, 5-HT transporter, and a range of genes for cannabis. So real statistical GE that may be pointing at biological interaction. So that's a quick tour. What have we done? We've looked at definitions of gene-environment correlation and gene-environment interaction. I've given you lots of caveats that the idea of environment is complex for psychiatry and really interesting, I think. We're looking at polygenetic disorders, so it's a more complex genetic field to look at interaction from when we were thinking of candidate genes that led to some of our key papers. But those key examples are classic examples of gene-environment interaction for those three phenotypic areas. But there are caveats, and I haven't really gone into the methodological background, but I've just highlighted some of the main problems. So, to conclude, I think gene-environment interactions in psychiatry are inevitable, and we're probably just scratching the surface of what's going on biologically. So you're coming into psychiatry at a really interesting time. Here's a little further reading, and I look forward to answering some of your questions in the next section. Thanks very much.
Hi, Peter. Can you hear me okay?
I can hear you, yes.
Yes. Thank you so much for such an interesting talk. It was really, really good. We currently don't have any questions in the chat yet for this talk. So if anyone has any questions or wanted to ask Professor Peter Jones anything, now is a great time. I'll start off with a question that I had. I just wondered whether the research on gene-environment interactions and which genes increase someone's sensitivity to environmental stresses, whether that knowledge could be used in a preventative way to sort of, whether testing in early childhood, if someone has a gene that increases their sensitivity to a stressor, whether that could be used in any preventative way.
That's a good question. And I think the honest answer is that at the moment, we're not in a position to say that we have strong enough evidence of large enough effects in GE that we can use them preventatively. There has been discussion about the cannabis example, and as to whether there could be genotyping that would guide largely young people, not only for cannabis, but for other drugs as well. But in a sense, we all know that if a group of young people, 12 people smoke cannabis together, then 2 or 3 of them might get quite paranoid, and the rest don't. And that's probably, or it could well be, an expression of some sort of vulnerability, and perhaps that's genetic vulnerability. So at the moment, I think it gives clinicians a framework in which we can talk to patients about why they might be vulnerable to this and that. I mean, in those examples, I suppose those classic papers in Science by Caspi, they came at the end of a time when we were still thinking that there might be specific genes that are particularly involved in specific disorders, whereas what the GWAS era has given us over the last decade is that we know that there's hundreds of genes interacting. So at the biological level, it's difficult to think of, single gene effects that we might test for in practice. But nevertheless, who knows? It may come.
Yeah. Okay. Thank you. Our next question, we've got a few now, is from Ahmed A, who I will just unmute now. I don't know if you're unmuted. If not, I'll just read out the question. I'll read out the question. The question was, how does epigenetics play a role in mental disorders?
So that's a very good question. And if you remember, Benjamin, when you put the title of the talk on Eventbrite, you put epigenetics behind a colon. And I said, take that out because I don't know anything about epigenetics. So I'll just give you that caveat. But 2 parts of the question. So I kept on banging on in the talk about those studies showing statistical interaction. So you do statistical tests and it looks as though the effect of one thing on another is magnified depending on genotype. So it's an inference that that is representing a biological process. However, if it really is representing a biological process, then there must be, one assumes that there is an epigenetic mechanism, that the cumulative exposure to those environments will be moderating either the long-term expression of a gene or the likelihood of a gene being expressed in a certain set of circumstances. So some of them will involve epigenetic mechanisms, and I guess particularly for the early life examples. And again, you'll be thinking about immunity in the next talk. So there might be environmental effects that prime immunological systems to react in a more exaggerated or minimal way on re-exposure later in life. And there's going to be epigenetic mechanisms involved in that. But really what my talk's been about is statistical studies to point the finger at biological mechanisms, and one presumes that those mechanisms will involve epigenetics.
Okay, thank you. The next question is from Jedwin Stanley, who I will try and unmute now. Again, it should work now. Jed, if you'd like to ask your question.
Cool, hello. First and foremost, thank you, Professor Jones, for that talk. It was very interesting. I was just wondering, do you ever see genetic testing becoming routine in clinical practice alongside like history taking and examinations, like just a standard examination?
I think I could see it becoming, at the moment, just to say what I've said again, what looks as though it's happening is that things are just actually becoming more and more complex. But I could see there being genetic testing in the clinic in your career rather than mine. However, I have begun to talk about this with patients. So I mentioned, but didn't really go into this concept of a polygenic risk score. So what's happened as we've got so many hits for polygenic chronic disorders like schizophrenia and depression, that we say there's 120 odd genes or more involved, then we summarize the liability in terms of an overall score. So you can simply take a score of alternative alleles across many genes and differentiate people in terms of a summary polygenic risk score. So you have a high overall score depending on your alleles of those genes, or a lower polygenic risk score. So it's a sort of continuous measure. And in fact, I'm dealing with a family now where their daughter committed suicide, and it looks, trying to reconstruct what on earth happened, that she may have had a certain psychiatric disorder. I won't go into what it was to protect their confidentiality. And in order to understand why that particular child in their family developed that disorder and the others seem to be mentally very healthy, they have been able to get a polygenic risk score calculated from tissue from their deceased child, and that, if it comes back very high or it comes back very low, that helps them understand why perhaps that child developed the psychiatric disorder whereas others didn't. But that's of no sort of practical influence for the patient, but it's been very useful for the family.
Great, thank you.
I mean, I think it's a different question. It's certainly different in learning disability syndromes. and that's a different talk really, but we are more able to get a molecular diagnosis for learning disability syndromes. And I think that will come in more and more for common disorders like schizophrenia and depression, that we'll see them fragment into a large number, or part of them may fragment into a large number of rare disorders. And those will be genetically identifiable.
Okay, thank you so much. The next question is from Allegra, who I will unmute now. If you would like to ask your question. Sometimes it takes some time. I will just ask I will ask the question now. It says, this might not be specific enough, but I'm interested in how to untangle the possible second peak in schizophrenia in women at menopause.
Well, I can't really untangle it for you either, although it really does seem to be real. A small peak around the menopause, and the mechanistic story is that that's due to reductions in estrogen, and that there is an interaction between estrogen biology and dopamine. But I don't think that it's fully understood. And actually it's an assumption that that peak from about 45 to 55 in women, you know, it's certainly highly likely to be due to the menopause, but it's not really understood mechanistically yet, as far as I know. Be interesting to look at hormone replacement, wouldn't it? I wonder whether there's data from large trials of HRT looking at onset of schizophrenia or similar disorders. Good question.
Yeah. So the next question is again from A. Ahmed. I will try and unmute you again. It doesn't look like it's working, so I will just ask. Are there any genes identified in people who thrive in adverse childhood experiences?
Sorry, could you say that again? The sound dropped out a bit.
Yeah, sorry. Are there any genes identified in people who thrive in adverse childhood experiences, or I guess in children who, yeah.
Thrive, was that the word?
Yes, sorry, thrive,
yeah.
Yes.
Okay, that's again a very good question. So this is really about resilience, or the concept of resilience, which is itself quite a contentious term. And I don't know of particular genes, but that would be a prime hypothesis. That comes back to the point, I suppose, from that Caspi study about what happens in very good environments. Well, the obverse is what happens when people seem to be resilient. And I guess it's going to be a polygenic answer, that there may be certain combinations of genes that make one resilient to adverse environments. But as far as I know, we don't, none of those have been identified yet.
Yeah. Okay, thank you. The next question is from Allegra, who I will unmute now. It doesn't seem to be working. I'll read it out. It said, also in the 5-HT case, can it be seen as similar but reverse as some CYP-XXX genes, tolerance for alcohol, and then heightened likelihood of alcoholism?
Yeah, well, that would be another gene-environment interaction. It's a nice example where alcohol is the environment.
Yeah.
So going back to the resilience question, and again, I keep coming back to this point that those studies, they're statistical studies about probability. So even in the case where, say, this 5-HT genotype that seems to magnify the effect of adverse life events. Within that group, it's only a probability that you have a higher likelihood of depression. There will be some individuals who have escaped depression. So it isn't a simple, it's not like a biological switch, it's a matter of probability pointing towards a biological mechanism.
Yeah. Okay. The next question, the person who answered it, who asked it, I can't seem to find. It's not completely on gene-environment interactions, but it is. Are there studies on the effect of psychotherapy on adult psychiatric patients who had traumas and adversities in childhood, compared to adult patients with similar stories who didn't have or receive psychotherapy?
So I don't quite follow the question. So would you mind repeating it?
Yeah. Are there studies on the effects of psychotherapy on adult psychiatric patients who had traumas and adverse childhood experiences, compared to adult patients with similar stories who did not receive the psychotherapy?
Who didn't receive the psychotherapy as well? Do you mean in terms of outcome? I wonder.
I'm not sure, I guess so.
Yeah, I mean, I think the story with psychotherapy has been that the more you look for trauma, the more you find, but not everyone has trauma, and it's not simply that they are recalling it. Psychotherapies develop, there are different flavors of psychotherapy, and I think cognitive behavioral psychotherapy, for instance, has developed to begin to encompass dealing with early life trauma. You can't simply look at depression as a set of current cognitions, the underlying, so-called underlying schema that might lead to that cognitive style associated with depression, with depression will have been, in some cases, influenced by early life events, some of them traumatic, some of them will have been supportive, no doubt. So psychotherapies are certainly, modern psychotherapies are trying to reach trauma in a way that they weren't doing 20 or 30 years ago, but not in the same way that classic psychotherapies in an analytical style began with trauma. So, interesting question.
Right, so not sure what happened to Ben there, but Ellen asked, are genetic correlations in polygenetic disorders like schizophrenia about particular symptoms or expressions of a disorder, like delusions versus hallucinations, or are they typically about disorders as a whole?
Again, a really good question. So I talked about endophenotypes and intermediate phenotypes. So this is where a measured phenomenon is perhaps closer to genes, closer to the transcription of a protein. And in schizophrenia, for instance, I suppose there is some evidence that the so-called negative symptoms might be more genetic, and those would be syndromes dominated by cognitive problems, and that those clusters of cognitive endophenotypes. But you know, the answer isn't really clear. So all those wonderful collections of cases that are going into the Psychiatric Genetics Consortium for schizophrenia, depression, etc., they are simply cases of schizophrenia or cases of depression, and the bulk of them were collected, or many of them were collected some time ago, and information about those clinical differences, hallucinations, delusions, negative symptoms, they're not really available to those GWAS studies. So it's a big limitation in the field, actually. And there's a lot of effort going on to try and collect additional cases where there is that phenotypic information. So what you're effectively doing is stratifying your GWAS study according to phenomenological characteristics. So very interesting question, not yet clear. The methods have got a long way to try and catch up. While people are waiting, I should point out that when I said I was showing you a map of England, it was, of course, a map of England and Wales. And my own forebears would have been shocked at me missing that out. And twin studies are part of the classic genetic methodologies, family, twin and adoption studies.
Brilliant. Thank you very much for coming. Unfortunately, Ben seems to have cut out. He was going to say a big thank you as well, I'm sure. So, yeah, I don't think Ben's coming back. So I think I'm going to introduce the next talk. But thanks again, Professor Jones, for coming and for taking the time out. Really, really interesting and exciting to see what the cutting edge of psychiatry looks like at the moment. Brilliant, thank you very much. I'm just going to get the next speaker up. Brilliant. Sorry, it's just loading. Okay, so our next speaker is Professor Edward Bullmore, and he's going to be talking about the inflamed mind, a radical new approach to depression. Professor Bullmore is a world expert in neuroscience and mental health. He has been a professor of psychiatry at the University of Cambridge since 1999. And in 2005, he also became the vice president of experimental medicine at GlaxoSmithKline, whilst maintaining his post at the University of Cambridge. Professor Bullmore recently published a book called The Inflamed Mind, which will form the basis of this talk, discussing the new science on the link between depression and inflammation of the body and brain. Thanks very much.
Well, hello, and thank you very much for inviting me to contribute to your mini conference for students, psychiatry and neuroscience. I think it's a really great idea to bring together the science of the mind and the science of the brain, and I hope you're all enjoying the conference so far. I'm going to talk about the inflamed mind. These are my disclosures, just so you know where I'm coming from, where I'm employed, what my sources of research support are. I don't think there are any major conflicts of interest, but just so you know. A lot of what I'm going to talk about is written at a fairly general level in this book, The Inflamed Mind, that I published a couple of years ago. And the book ends with these few sentences. We could be on the cusp of a revolution. It won't be televised, and I might be wrong, but I think it has already begun. And that probably sounds a bit cryptic, but hopefully it will become clearer over the course of the next 35 minutes or so. I wanted to start with a story about how I started in psychiatry, which is quite a long time ago now. It's about 30 years ago. I found myself at the Maudsley Hospital in South London, which is quite a grand facade and had a tremendous reputation at the time as the sort of training ground for British academic psychiatry. So I was pleased to find myself there. This is a picture of me about 30 years ago, looking pleased with myself at the Maudsley Hospital. And it wasn't very long before I started working in the outpatient clinic. And I met a patient not much older than me at the time who came along and said, you know what, I'm feeling pretty blue, pretty sad. My sleep's not good. My appetite's down. I can't remember when I last had sex. I'm feeling guilty about things I've done in the past and pessimistic about the future. And I looked at him and said, well, I think you might be depressed. And he was very unimpressed by that remark. He said to me, well, I know I'm depressed. That's why I asked my GP to refer me to the Maudsley Hospital. The question is, what are you going to do about it? And I said, oh, well, this was 1990, remember. I said, well, you know, actually it's a very exciting time for treatment of depression. We've got these new antidepressant drugs, like Prozac, you might have heard of, that boost levels of serotonin in the brain and set right this sort of transmitter imbalance that we think causes depression. And he looked at me and said, how do you know that's true? And I said, well, I've just been reading about it, actually. And he said, no, how do you know that's true about me? How do you know that there's an imbalance of serotonin in my brain and that these drugs you're offering me are going to work? And it was a very powerful moment for me because I realized immediately, and I think he did too, that I didn't have an answer to those questions. I had no evidence that there was an imbalance of serotonin in his brain. Even now, 30 years later, we don't know anything about the status of serotonin in the brain or the body of patients that are offered treatment with serotonin reuptake inhibiting drugs, so-called SSRIs. So it was a very powerful moment because he pinpointed actually rather an important area of ignorance in modern psychiatry. And I must confess, he did, when he left the clinic, leave me feeling a bit of a fraud. He left me feeling like one of these old-fashioned 18th century physicians who might have treated depression as melancholia, a disturbance of the black bile, this mysterious substance that circulated in the body but was never measured. It seemed to me that actually offering treatment with serotonin-boosting drugs without really knowing what the status of serotonin was not that far from the old school of Hippocratic medicine that we thought we'd left behind. So those thoughts have sort of stayed with me for many years, as you can tell. And if we look now at where things stand in the terms of the treatment of depression, it's not all bad. These are results from a recent meta-analysis. And just to orientate you, each of these little blue squares indicates the treatment effect for an antidepressant compared to placebo in a large number of clinical trials. If these drugs were not any better than placebo, then the confidence intervals, these horizontal lines, would touch the vertical line of no effect. But you can see that they don't. They're all shifted to the right. That means that for all of these drugs, there is robust randomized clinical trial evidence of significant efficacy compared to placebo in terms of reducing the symptoms of depression. So that's good. We've got a number of drugs which work as antidepressants, as ways of relieving the symptoms of depression. But there are still a number of caveats. We don't know that the serotonin levels or noradrenaline levels or dopamine levels in the brain are at a low ebb in patients who are offered treatment with these drugs. Partly for that reason, we don't know which patients are most likely to respond well to any particular antidepressant drug. And it's worth noting that although there are significant treatment benefits on average, which is what the data are showing us on the left, there is a large minority of patients with depression who don't experience any meaningful benefit after treatment. So-called treatment resistance depression accounts for about a third of cases. There's been very little therapeutic progress. If there had been a lot of therapeutic progress, this drug up at the top with the biggest difference from placebo would be the most recent drug. But as you can see, it's amitriptyline, a drug that's been around in psychiatry since the early 1960s. It's one of the oldest drugs we have available, and yet still one of the most effective. So that's not a sign of progress. And, you know, while therapeutically we may not be moving forward very fast, depression remains a considerable problem. In fact, a lot of people estimate that it is on course to become the single biggest cause of disability worldwide by 2020. Now, you might have thought that in that context, where you've got a big problem with depression and not much evidence of recent therapeutic progress, that drug companies and other investors would be piling in to find breakthrough new treatments. But actually, industry investment in R&D for depression has stalled somewhat since about 2010. That's an industry-wide trend that came home to me very acutely when I was working half-time for a pharmaceutical company, GlaxoSmithKline. And my boss at the time called me up one day and said, "We're out of psychiatry. We're not investing in any new antidepressants. We can't see a way to make it work as a business." And I said to him a few weeks later, "Well, you know, where does that leave me, you know, as a psychiatrist in a company that doesn't want to do psychiatry anymore? Can you imagine any hope that we might get back into antidepressant development in the future?" And he said to me, "Well, I'd never say never, but if we were going to do it, it would have to be in a completely different way to the way that we've done. We thought about depression and we've thought about developing antidepressant drugs in the past." And that, again, for me was a very powerful moment, a moment when I began to think quite differently about depression. And one of the ways in which I began to think about depression following that conversation in relation to inflammation. Now, inflammation, we all know, is important in terms of the body's response to trauma. And in this cartoon, you can see there are a couple of cavemen having a fight. One of them is wounded in the hand, and a few days later, it's red, it's swollen, it's tender, he can't use it. Those are the classical signs of inflammation. We've known about that, those signs, since the Romans. We've also known for a long time that under the skin of this sort of clinical change, there's a lot going on in terms of cellular and molecular activity. So here's the blade of this spear penetrating the caveman's skin. You can see the blade is covered with bacteria. So this is an infectious wound. And the body's immune response, shown here by these little schematics, these are the macrophages or innate immune cells that sit everywhere in the body waiting to detect evidence of trauma or infection. They're the first-line troops, if you will, that respond very quickly to this trauma and begin to ingest the bacteria, destroy them by phagocytosis, and release inflammatory hormones or cytokines that circulate through the blood and encourage other innate immune cells to leave the peripheral circulation and join the fight against infection at the site of the wound. Now, that is sort of a very basic account of innate immune response to a local trauma. But as we've learned more about immunology over the last several years, it's become clearer and clearer, I think, that inflammation is never really local. There's always an important systemic component to it. So there are many components of the immune system throughout the body, as you know, lymph nodes, the spleen, the bone marrow, the thymus, and so on. And they are all coordinated with each other in a very complex, intelligent, adaptive system. So macrophages that pick up evidence of infection in the periphery can carry that antigen into the lymph nodes, for example, where they come into close contact with lymphocytes, which in turn produce antibodies that circulate back to the site of the injury to facilitate the immune response. So as we've learned more about the immune system, we've realized how important it is to think of it as a system, and how important it is to recognize that inflammation, even if it looks local, in terms of swelling and redness in the hand, say, is always the tip of an iceberg, and there is a systemic immune response going on under the surface. Now, how does that relate to depression? Well, the first patient that I can remember seeing who was both inflamed and depressed was not in a psychiatric clinic, she was in a medical clinic. This is, again, going back a few years. About a year before I started at Maudsley, I was still training as a physician, and I met a woman like this with rheumatoid arthritis in a medical outpatient clinic. And as a young physician at the time, of course, I focused on the physical signs of arthritis, the swollen joints, the deformities. But I soon noticed that she was feeling or looking quite sad, and I asked her some questions, and she told me very fluently and quietly, more or less exactly the same as the patient that I would meet later at the Maudsley. She too was feeling sad, her sleep was disturbed, her energy was very low, she had difficulty thinking clearly about the future. So again, she seemed to me quite clearly depressed. And I remember going to the consultant physician in charge of a case and saying, you know what, I think I've made a discovery about this patient. Not only does she have arthritis, she's also depressed. I was quite pleased with myself at this diagnostic discovery, but he looked up briefly and said to me, well, you would be, wouldn't you? And what he meant by that was quite complex, really. He meant her depression is a psychological reflection on her physical state. She's looking ahead, she's thinking about the years of disability in the future. What he was ruling out is the possibility that the same systemic autoimmune process that was causing her swollen joints might also be mechanistically driving her mood change. And one of the reasons that he was so quick to rule out that alternative explanation is that at the time, and this is again 1989, the dogma was that the brain and the immune system had nothing to do with each other. The brain was so-called immune privileged, which meant that the inflammatory proteins and cells that might be passing through the blood stream in the brain could not access the brain itself. The blood-brain barrier was conceived as something like the Berlin Wall, an impermeable barrier. So the only possible cause of her depression had to be either what was going on in her mind or what was going on in her brain, but it could not be what was going on in her immune system, particularly her peripheral immune system. Now, looking back on that, it's remarkable how much has changed in terms of our basic knowledge about how the immune system and the nervous system relate to each other. It is certainly not the case any longer that we can think of the blood-brain barrier as anything like the Berlin Wall. In fact, a lot of people prefer to call it the blood-brain interface rather than the blood-brain barrier these days, because we know that there are, in fact, many channels of communication by which cells and proteins and other inflammatory signals can get from the blood and into the parenchyma of the brain. And once those inflammatory signals have reached the brain, they can activate the brain's own immune cells, the microglia, which are very closely related to monocytes and macrophages, sitting in the brain, can pick up peripheral inflammatory signals, themselves become activated, produce locally high concentrations of cytokines and other inflammatory molecules, which in turn change the function of nerve cells. Nerve cells don't like, generally speaking, don't like being in an inflamed milieu. For example, if you expose nerve cells in culture to cytokines, it changes the way they metabolize serotonin. It changes the way that serotonin uptake transporters are expressed on the surface of nerve cells. So there is in fact a sort of mechanistic pathway from peripheral inflammatory immune cells and proteins through the microglia that could affect neuronal function, ultimately cause change in large-scale brain circuits that we know are important for emotion, and so cause depression. Now, if that was true, then anti-inflammatory drug would be antidepressant. And actually, there's quite a lot of evidence for that from trials of anti-inflammatory drugs for inflammatory disease like rheumatoid arthritis, SLE, and other autoimmune disorders where depression, fatigue, cognitive impairment are very common comorbidities, so-called. And what we did in this study was to go back and look at about 12 randomized control trials of anti-inflammatory drugs that had been conducted using or enrolling patients with inflammatory disease. And as part of the assessment protocol, all of these patients have been asked a few simple questions about their mood. And that wasn't the purpose of the trial. It was a sort of what we call a secondary endpoint or an exploratory endpoint that was measured in each of them. And in this analysis, we went back and we looked at all of those data and we said, well, let's analyze the data as if those mood scores had been the primary endpoint, the main point of the study. What evidence is there that anti-inflammatory drugs have effects on those measures of mood? And what this meta-analysis is showing is that there's a fair bit of variability in terms of the treatment effect. But on average, over all of the studies, you can see this little diamond down at the bottom is clearly separated from the vertical line of no effect, meaning that these drugs on average have significant antidepressant effects compared to placebo. In fact, the effect size for all of these different drugs on average is about 0.2, which is not that far from the grand average effect size of serotonin inhibitors and other monoaminergic antidepressants. Now, people are quick to say, well, that's because when you give an anti-inflammatory drug to somebody with rheumatoid arthritis, their physical symptoms improve and therefore their mental state picks up as a consequence of that physical improvement. We can try and disentangle that by looking at a subset of the patients in the trials who did not respond physically to the treatment and ask, in those patients who remained unwell in terms of their physical symptoms, was there nevertheless any evidence of improvement in mental state? And the answer is yes, there is. So even in those patients who did not improve physically, we're seeing a significant antidepressant effect of these anti-inflammatory drugs. Now that's, I think, very encouraging and it needs further follow-up. But of course, we're talking here about so-called comorbid depression, depression that arises in the context of a major inflammatory disease like rheumatoid arthritis. But as psychiatrists, what we mostly are concerned with most of the time is major depressive disorder. And these are the DSM-5 diagnostic criteria for major depressive disorder, which you may already be familiar with. Just to remind you, there are two cardinal symptoms of depressed mood and loss of pleasure. And then to have a diagnosis of MDD, you need at least 3 of these additional symptoms, which are quite vague. So weight increase, weight decrease, sleeping too much, sleeping too little, all of these are symptoms of depression. They must collectively cause significant distress or impairment, and they must be present for at least 2 weeks. And that is how we diagnose MDD in clinical practice. What's often forgotten is that in addition to those inclusion criteria, there's quite an important exclusion criterion. The episode must not be attributable to a substance or a medical condition. So strictly speaking, by definition, somebody with a diagnosis of MDD can't have an inflammatory condition going on in the background that might explain their symptoms. That would technically rule out a diagnosis of MDD. In the book, I talk about this as an example of medical apartheid, the hard fault line that I think still runs through the middle of Western medicine, separating the way we think about the mind from the way we think about the body. It's an idea that goes back at least as far as Descartes. This is one of the beautiful diagrams in Descartes' treatise on human nature. And of course, he very elegantly showed how we could think about the body in terms of physical mechanisms and mathematical laws. But he supposed that states of mind, like the mood, were qualitatively different and could only interact with the body through some nebulous mechanism that he located in the pineal gland. That seems kind of quaint and absurd in detail, Cartesian dualism, but in principle, it's still very characteristic of mainstream medicine. And yet, I would argue it's out of date. Why do I say that? Well, because if you look at patients with major depressive disorder, if you measure in blood the levels of inflammatory cytokines, there's pretty robust evidence of low-grade inflammation quite consistently. So again, this is a meta-analysis, and here we're looking at the difference between depressed cases and healthy controls in terms of interleukin-6, which is one of the most important pro-inflammatory cytokines. This vertical line shows that there's no difference between the cases and controls, and here are the data. And you can see that there's an effect size of about 0.5, indicating a significantly increased level of IL-6 in major depression, also significantly increased levels of C-reactive protein, tumor necrosis factor alpha. Those are the cytokines most consistently implicated. Now, people look at those data and they'll say, well, yes, that's a very robust association, but it doesn't prove causality. How can you be sure that these inflammatory conditions are actually causing the depression? So that's not an easy question always to address in humans, but one way of thinking about it is in terms of the temporal order of things. You know, we know that cause precedes effect. So if inflammation caused depression, we'd expect to find examples of inflammation preceding depression. And in fact, there's quite a lot of evidence for that over a number of different timescales. So if you have a typhoid vaccination, for example, that induces an inflammatory response. And for many people, it'll also induce 24 to 48 hours of mild depressive symptoms, so-called dysphoric symptoms. On a slightly longer timescale, patients with hepatitis C used to be treated with interferon alpha, which is a powerful pro-inflammatory cytokine. But it had a significant complication. About a third of those patients developed major depressive disorder some weeks later. And the data here indicating an example of inflammation preceding depression over an even longer time scale. So these are data from a longitudinal sample of young people who had blood measured for IL-6 at the age of 9 and were then assessed for depression about 10 years later. And what you can see is that the children who were in the bottom third, the lowest tertile of the distribution for IL-6 at the age of 9, had lower rates of depression than the children who had IL-6 in the top third of the distribution at the age of 9. So in this case, low-grade inflammation detectable by a blood test is preceding the emergence of depression by about a decade. So whether it's over the course of days, weeks, or years, inflammation can precede depression and therefore could be causal. Let's turn now to the question of what would cause inflammation that in turn causes depression. And there are many bodily factors that could be relevant: obesity, the gut microbiome, menopause, aging, winter. All of these factors are associated with increased low-grade but increased inflammation. Most interestingly, from the point of view of psychiatry, to my mind, is the evidence that it's not just bodily factors that can drive low-grade inflammation, but also social factors, particularly stressful situations, bereavement, caring for a dependent elderly relative, public speaking, or childhood adversity. All of these stresses have been associated with inflammation, and of course, many of them, particularly childhood adversity, are well-recognized risk factors for depression. This is a little bit more data showing that in a large cohort followed up over many years, the individuals that had experienced more than 2 adverse childhood events, so maybe abuse or poverty or emotional neglect, those deprived children, when followed up as adults, had significantly increased levels of CRP, a blood protein marker of inflammation. So this association between stress or maltreatment in childhood and later inflammation seems to be quite robust. And those sort of data allow us to sketch out this sort of scheme. It's here shown very much as an artist's impression, but inflammation, whether it's driven by obesity, arthritis, whatever might be the cause, can communicate with the brain, can cause changes in brain function, which in turn lead to changes in mental state that we recognize as depression. Becoming depressed may itself increase the risk of social stress, but in any case, social stress itself is another potentially primary cause of inflammation that drives this whole vicious cycle. That looks like a caricature. It is a caricature, but it represents in simple terms a much more detailed mechanistic understanding of how stress through the sympathetic nervous system can cause a change in the peripheral immune system, which in turn can communicate across the blood-brain barrier to change the way that neurons are functioning. So there is quite a bit of biological detail now available in support of this general idea. We still have to ask ourselves the question why inflammation might cause depression, and the answer to that has to be something to do with evolution, has to be something to do with natural selection. There must be some sense in which it is advantageous or was advantageous for the inflammatory response to be able to co-opt behavior. And people have come up with sort of theories for this. So it's been proposed that this caveman might have found it easier to heal the wound to his hand if he'd been socially isolated and subdued, had been less physically active, had perhaps changed his diet in some way. In other words, you can make up a story that some of the symptoms of depression could have had survival advantage as part of a whole body, whole mind response to trauma or infection many, many years ago in our evolutionary history. Now, if that was true, you might expect that the genes for depression, which we are just beginning to discover, might be genes that are specialized for immune functions. And this is a Manhattan plot, as you probably recognize, showing the whole human genome laid out on the horizontal axis. And the height of the skyscrapers above the x-axis indicates the significance of association of particular genes with depression. And I think it's very exciting for psychiatry that we are finally beginning to see genes emerge from these kind of genome-wide association studies that are statistically robust. And these are data recently published for a large study of major depressive disorder, highlighting a number of genes that might be interesting to consider as possible agents in causing depression, but also as genes that have an important role to play in the immune system one way or another. And this is still very early days, but I was very struck just looking at this quite casually that This gene here is olfactomedian 4. It's highly significantly associated with depression, and in mice, at least, it does play an important part in the immune system. It regulates the immune response of the stomach to infection. So I think this is very, very early. I'm not trying to convince you that there's a clear genetic story about inflammation and depression yet, but the data are becoming available, which will allow us to investigate that theory in more detail in years to come. So what? So, I've tried to persuade you that we should think about inflammation and depression as robustly associated with each other, as evidence of causality, and then maybe even the beginning of a genetic linkage between inflammation and depression. But how is that going to help us in terms of treatment? Well, as I've already said, if inflammation causes depression, then anti-inflammatory drugs might be expected to treat depression. Could anti-inflammatory drugs emerge as a new class of treatment for depression in psychiatry? I would say maybe, but not yet, and never for everyone. I think what's more likely, and in many ways much more exciting, is the idea that in future, people come to see us in the clinic, we recognize that they're depressed, and then we do a test, we measure a biomarker of some sort to identify whether or not they might have an inflammatory cause for their depression, and then make a treatment decision on that basis. And if the biomarker indicates that they might benefit from an anti-inflammatory drug, well, then that would be the course of treatment that we would offer. And otherwise, there are many other treatments for depression that might be more effective. That sounds very basic, and it is pretty much motherhood and apple pie in the rest of medicine. You're not trying to treat the symptoms, you're trying to treat the cause, and you're using investigations to find out what that cause might be. But that sort of model in psychiatry would be a big advance on where we've been for the last 20 years or so. We will need biomarkers. I know that because when people have tried to treat major depressive disorder with anti-inflammatory drugs in clinical trials like this one, which is a trial of an anti-TNF antibody in patients with MDD, there is no significant benefit of treatment over placebo in this study. When you look at the data overall, it's only when you dig a little bit deeper and look at the relationship between treatment response and baseline levels of CRP in the depressed patients that it begins to look a bit more positive. In this study, the patients with high CRP levels benefited from anti-TNF antibodies. The patients who were depressed but not inflamed did not benefit from that treatment. So that, I think, is telling us we need biomarkers, we need to develop tests, we need to be much clearer about which patients with depression might be inflamed in a way which is open to treatment. I don't know what those biomarkers will be. I don't think anybody yet knows, but I don't think it's going to be a single protein like CRP or IL-6. My bet for a peripheral biomarker, a blood biomarker, would be some sort of gene expression network. And these are data just showing in a case control study of patients with depression compared to healthy controls that the significant increased expression of a network of genes shown here in green that are specialized for innate immune function, and reduced expression of another network of genes specialized for adaptive immune response. I think these kind of transcriptional network changes are a much better bet for biomarkers of the future. Two final points. I've talked about drugs as interventions, particularly anti-inflammatory drugs as new treatments for depression. And that reflects my own personal story. How I came into all of this was from a pharmaceutical perspective and thinking about the development of new antidepressants. But the basic idea that depression and inflammation might be causally related to each other allows us to think about non-drug interventions. Vagus nerve stimulation, for example. This is an intervention that has been licensed for depression in the States for, I think, about 15 years. Nobody's ever really understood how it works. But one interesting piece of new science that's emerged in the last couple of decades is this so-called vagal inflammatory reflex, which makes the point that the vagus is sensitive to peripheral levels of cytokines, so it's sensitive to the inflammatory state of the body. It can signal that information to the brain, and then it can produce an efferent output to the spleen, which damps down the peripheral production of cytokines. So it's a homeostatic circuit of exactly the same type in principle that the vagus supports in relation to, the heart, the lungs, the gut, all homeostatically controlled by the vagus to some extent. The same seems to be true of the immune system, and conceivably stimulating the vagus nerve might have anti-inflammatory effects, which in turn explain its antidepressant effects. That's possible, and at least it's not a drug. The other point I'd make is it's not just about depression. It could also be these ideas of neuroimmunology, of brain-immune interaction, could be relevant to many other neuropsychiatric disorders. This is the first patient with Alzheimer's disease. When Alzheimer looked at her brain under the microscope, he famously noted these plaques of abnormal material. We now know are amyloid and plaques. But we've also recognized that increasingly that the brain may see these abnormal proteins as antigenic. They may stimulate a local immune response. And in this slide, you can see the plaques are surrounded by microglia, the brain's innate immune cells. So there's a lot of interest, not just in psychiatry, but in neurology also, about the implications of the immune system for understanding the pathogenesis and progression of neurodegenerative diseases. So with that, I'll finish. I'll take you back to where I started. This book, if you're interested in the area, offers a general introduction, provides some references to more specialized scientific papers that might be of interest. We could be on the cusp of a revolution. By that, I mean, we could be on the cusp of moving beyond the dualist tradition that has endured in medicine for the last several hundred years. It'll be a slow process. That means it won't be televised. It'll take several generations. It's probably something that your generation will see more clearly than mine. And I might be wrong. Most of what I've told you is pretty substantial and supported by a lot of evidence, but I've also indulged in some speculation, for example, about the possible genetic mechanisms that might link inflammation and depression. So, this is science evolving. Not everything we think we know will turn out to be right. But I do believe that we have already begun on what I think is a very exciting journey for medicine, and particularly for psychiatry and the relationship between the mind and the body in how we think about depression. So thank you very much.
Thank you very much, Ed. I can see you're here. So I think, brilliant. I can see you. Just unmute you now. Brilliant. Can you hear me okay?
Yeah, I can hear you fine.
Oh, yeah. Thank you so much for attending tonight and for pre-recording that talk. You have this incredible ability of explaining such complex concepts in a way that everyone can understand. So thank you so much.
Thank you.
So we've had a question from Sophie to start off with. She's asking, would you hypothesize that talking therapies would reduce inflammation?
You can see how that could happen, yeah. I think there is a bit of work showing that, in fact. Mindfulness interventions have been shown to have some immune calming effects. I would say it's still, what I think about it is it's conceptually possible. I would like to see it kind of shown a bit more clearly. But there are some encouraging preliminary data.
So what do you think is the next step with looking at that kind of research then? Because I know they've done some studies on, obviously originally it's antidepressants and therapy, and then they've done some on psilocybin and therapy. Are you thinking that it could be anti-inflammatory medications and therapy as well?
Or that, psychological interventions, behavioral interventions like yoga, meditation, these might also have immune modulating effects. I mean, a lot of people think they do. I think there's an opportunity to do more of that kind of research where you use a psychological or a behavioral intervention, but you do measure the immunology.
Yeah.
You think about the immunology properly. I mean, one thing that does get me, I don't feel completely easy about, let's say, is if you look on Amazon, for example, there are a lot of books called things like Anti-Inflammatory Diet. And I've read a few of them. And I've never seen much immunology or evidence for an anti-inflammatory effect of these things. Now, I don't think that it's impossible that, certain diets, certain psychological interventions, or meditation, other lifestyle practices could change your immune system. And I think it would be interesting to do more of that research. But I think you've just got to keep, and now I'm speaking particularly to medical students, stay in touch with the immune system and make sure that you're actually seeing data on inflammation changing when people are telling you that these interventions might work by immune mechanisms. I think they could do, but we need to see the data.
Sure, thank you very much. We're going to try and get Deborah. She wants me to read it out. So she asked, do you think we should be overcoming this dualist tradition through having neurology and psychiatry training as a joint endeavour?
Well, I think that would be a good idea myself. I mean, I think, there's a bit too much of a split between neurology and psychiatry. In other countries, they do take a more integrated approach to training. So, for example, in Germany, I think it's much more commonly the case that your neurologist will have had a lot of psychiatric experience and vice versa. There's, I think there is an awful lot of common ground between them. Now, obviously, people are going to want to super specialize, as it were, on that spectrum ultimately. But I think certainly the core training could be a lot more integrated.
So what can you see happening then? Would it be psychiatrists specializing in immunology? Would we have immunopsychiatrists quite in each hospital?
Well, I mean, I think just having, just having more crosstalk between neurology and psychiatry trainees and having, I don't know how, certainly when I was, training as a psychiatrist, there weren't, I can remember one or two neurologists, doing even 6 months of psychiatry as part of their training program, and vice versa. It wasn't very common. I think more psychiatrists had done a bit of neurology before they went to psychiatry. I did a bit, but not as a sort of neurologist, more as a sort of trainee physician. But that was always not, it was never a very, common cross current. People got separated quite early. In fact, straight out of house jobs, more or less, people got segregated between psychiatry and neurology, and there wasn't that much crosstalk. So I think, I think things could be a lot more integrated without necessarily without the added, aspect of integrating neurology and psychiatry, for example, in terms of, immune mechanisms. I think if you just sort of swap people around the training programs a bit more, that would probably be a good start.
Yeah, thank you very much.
I'd also just briefly say the Royal College of Psychiatrists has just sort of taken steps to update its training curriculum.
Yeah,
so it does have more neuroscience in it, and I think that's a very positive step.
Absolutely. So Amelia asked, could you explain how many patients with illnesses such as irritable bowel syndrome or chronic illnesses like ME or CFS effective overactive cortisol often have associated mood disorders? In terms of your inflammatory kind of hypothesis.
Right. Well, I mean, so I don't know is the honest answer. I don't think anybody really knows exactly how many of those syndromes are caused. They're all of them quite mysterious. They all sort of sit somewhere sort of on the interface between mind and body. And that means they're poorly understood because, you know, the things that a dualist system doesn't see very well are the things that sort of sit at the interface. I mean, I think immune mechanisms, you know, could be relevant. I mean, you know, the immune system is relevant to pretty much every disorder you can think of, one way or another. And, you know, this new science does allow you to think about ways in which, let's say, IBS, you know, could imagine that there was some sort of immune response in the gut, a sort of a change in bacteria, microbiome, that might cause local gut symptoms. It might also cause a sort of low-grade systemic inflammation and a depressive response. There are also a lot of vagal afferents in the gut. The vagus, we know, is quite sensitive to local cytokine concentrations, local inflammatory state. So you can see how you could get an inflammatory signal going from the gut, and that might give you a syndrome where you had mood with, you know, typical irritable bowel type symptoms. I mean, that is, you know, as you can see, I'm waving my hands quite around quite a lot when I'm talking. So I'm just basically making that up. But I'm not really making it up de novo, but that's a sort of high level model that I think a lot of people are interested in. But again, it's going to, you know, need more data, more specificity, you know, and, you know, one would want to be sure that, you know, these mechanisms for irritable bowel, let's say, that we're talking about, could actually be measured in individual patients in real life. So we're not sort of talking in a sort of abstract way, like, you know, the serotonin theory of depression. We're not talking about something that, you know, we think could be, could conceptually be relevant, but we can't actually measure it in individual patients. So again, I think we just got to stay close to the data and particularly the immunology, and not get too abstract about it.
Thank you. Jazz asked, given that I think you've kind of answered the first bit. So they were asking a little bit about the links in terms of information between depression and in terms of eating disorders as well. Yeah. So such as bulimia.
Yeah. Yeah, I don't know much about that, actually. That's an interesting question. It's not, you know, it's not an area where I don't think there's a lot of work that's yet been done on that. Yeah, no, I don't think I've really got much to say about that, unfortunately. I mean, we know that, well, let's put it another way. We know that anorexia is easier because, well, it's easier to think about from a sort of physiological sort of point of view, because obviously you've got this severe change in body weight, which is going to have major effects on your neuroendocrine system. And those in turn could have effects on immune response. I think it is generally the case that people with sort of severe anorexia tend to have relatively reduced immune responsivity. I'm not aware of a strong story about inflammation and anorexia. I think it's more that the immune system is sort of, well, essentially starved in the same way that the rest of the body is. I mean, I don't know how much energy the immune system usually consumes as a sort of proportion of the body's total budget, but it's probably quite expensive. And so it will suffer under starvation conditions. But that's the only sort of connection I can think of off the top of my head.
Thank you. Grace asked, when I was I was taught that MDD was caused by neuron death. Is this wrong?
No. So one thing to come back to maybe is what we mean by caused by. Okay. So is there an association between MDD and neuronal death or damage? You know, perhaps not necessarily at the level of the whole neuron, but in terms of reduced synaptic density or other markers of neuronal unhappiness, if you will. Yeah, there is some evidence for that. That's an interesting association. But I think when you think about depression, in fact, most psychiatric disorders, you have to try and think on multiple different levels. So you've got a neuronal change, which is contributing to, connectivity in large brain systems that are important for things like emotion and behavior and, a sense of pleasure and so on. I mean, all the psychiatric symptoms, mental symptoms, cognitive symptoms, they're all going to come out of, large networks of many neurons. So you have to think at that sort of slightly larger anatomical scale, and also think, well, what's causing those neuronal changes that you can see? There could be many reasons for that. It could be, the outcome of a developmental process. It could be, a more rapid reaction to acute inflammatory states. That's possible. So I suppose the point I'm making really is that I don't think that allowing, opening the door to the immune system necessarily means you've got to sort of close the door to other things that we knew about depression or indeed other psychiatric disorders necessarily. It's just a different way of putting them in context. It's like I make the point in the talk a bit. If you ask yourself, well, what's the one thing that we really know causes depression? It's stress. And one of the things I like about the way the immune system surfaces in the story is it doesn't try and tell us, it's all about the immune system, it's nothing to do with stress. It says it could be through the immune system that stress has those effects. So I think it's more a case of trying to bring things together in a more integrated overall understanding than sort of, saying it's immune rather than neuronal or neuronal rather than social.
Yeah. I read a book on The Body Keeps a Score, Bessel van der Kolk. What's your view on that? Do you agree with what he says?
I don't know. I've never read it. I'm very, very envious of his sales. That book has sold millions.
Yeah. I thought you'd be interested since it's on that kind of topic.
Yeah. No, I think I'm too envious.
Okay.
If his sales and his Amazon ratings, I can't bring myself to read it.
Okay.
Seriously, I should. And the fact that it is such a an enormously successful book is, I think, telling us that there's a lot of interest in the sort of general population for ideas that link the mind and the body. I mean, I think most people think that's kind of common sense. That's lived experience, and it's only when they encounter the medical profession they realise that things can be seen a bit differently through the traditional prism. But I think I haven't read it.
It's okay. We'll move swiftly on. We'll forget I ever mentioned it. So we'll go, and this is the last question actually, Ed. So this is from Rachel. Oh, we've got two more actually. Rachel said, is there any evidence or could research be done into correlations between depression and the strength of one's immune system? And as a side note, she said, or correlations between vitamin D, for example, and depression.
Well, I think vitamin D has been tried. I mean, vitamin D is an intervention, so you can put that through a randomized controlled trial. I think those have been done for depression, and I don't think they've shown an antidepressant effect. Now, that gets us back to the familiar thing with treatment trials in depression, which is, that they're often not stratified samples. So, if you just go out and you recruit 200 people with major depressive disorder, they're not all going to be depressed for the same reason. Why would they all respond equally well to vitamin D or anything else? So, we've got negative trial data. Does that mean vitamin D could never work for anybody with depression? I don't think so. But I think if you wanted to, assert that more, certainly you'd need a trial which involved a sort of stratified sample. You'd have to have some biomarker that predicted vitamin D response, and that could conceivably be an immune marker. I don't know. So I think with vitamin D, at least, and other interventions, you can get to trials and beyond correlations.
In terms of when she's talking about the strength of one's immune system, has anything been done about someone with a stronger immune system is less likely to get depression, or there's an association between the two?
I don't think that has really been done. But I think, again, you'd have to sort of question. You have to ask yourself what you really mean by strength.
Yeah.
And what but when we talk, I mean, I'm not an immunologist. Obviously, I did a bit, and then I've been a psychiatrist and neuroscientist. So I still feel very aware that when I talk about things like inflammation or the immune system, we're really talking about a massively complex thing. And the strength of the immune system, I think you need to sort of say, well, what exactly does that mean, and how would you measure it before you could tackle the question? I mean, if you mean greater resistance to infectious challenge or inflammatory challenge, I mean, there are ways of testing that. From a blood sample, you can get cells out, you can expose them to challenges, you can see how robustly they respond. And people have begun doing those sort of assays, ex vivo assays, as a way of testing the sort of fieriness of the inflammatory response to a bacterial challenge, for example. So yeah, those things can be done, but I think, just think about trying to get down to the detail, because the more specific and clear these ideas can be in terms of, at the same level as sort of other medical theories, the more persuasive they'll be, and the more likely they will be to lead to new treatments, I think.
Brilliant, thank you. This is the last question. Timmy asked about autoimmune diseases and depression. Is there a correlation between the two?
Yes, there is. Quite a strong correlation. So just to take one example, rheumatoid arthritis. Yeah. As an autoimmune disease, and that's depression. Depression is very, very common, major depressive disorder, but also a lot of the patients who have autoimmune diseases like rheumatoid, they feel quite ashamed to have a diagnosis of depression. And there's much more talk about fatigue, brain fog, lack of energy, but loss of pleasure in life. People will avoid the D word, but there are a lot of psychological symptoms and cognitive impairments associated with rheumatoid and other autoimmune diseases.
And Timmy also asked, how do we identify the cause in closed feedback processes, or in cases where the detection of one event, inflammation, overshadows the other, depression?
Well, I mean, I think if I understand the question right, I think it is difficult to get away from us. There isn't necessarily a simple causal arrow that sort of starts at A and so it starts with inflammation, ends with depression. I think there certainly could be circular causality. You can imagine circumstances under which depression, change of mood, change of behaviour could cause changes in the immune system. It isn't necessarily a one-way street. And I think that is difficult to disentangle conceptually. I don't think it perhaps matters as much therapeutically, because, you know, whether it's a straight arrow or a circular arrow, you know, if you can interrupt it at any point, you could still break the effect, the relationship between you know, the two agencies. In this case, inflammation and depression. So I think there are some conceptual challenges in thinking about the circularity, as there are in, you know, relationship between, you know, depression, obesity, childhood abuse, for example. That's a very well-known constellation of syndromes, and there's a lot of talk about the causal relationships between those factors. So I think, yes, it is causally complex, but I think that doesn't necessarily mean it's therapeutically, you know, impossible to make progress.
Thank you so much. That's all the questions. Thanks so much for coming again.
All right, well, it's been a great pleasure. I've enjoyed the chance to meet you, and I admire your stamina and dedication. Going on
Thanks very much.
You know, late into the night like this. It's very impressive.
Thanks very much.
Okay.
Have a good evening.
Bye.
Cheers. So our next talk is on an autoimmune cause of psychosis. Dr. Venkatesan is a consultant neuropsychiatrist at Cumbria, Northumberland, Tyne and Wear NHS Foundation Trust. She has a keen interest in behavioural changes that follow brain injury. In her talk today, she will discuss some cases of autoimmune encephalitis. Thanks very much.
A very good evening to all. Thank you very much for inviting me to share my experiences of working with patients with autoimmune encephalitis with you this evening. My name is Gayatri Venkatesan. I'm a consultant neuropsychiatrist and I work in Walkergate Park in Newcastle. So this is Walkergate Park. This is based in Newcastle, and this is a Centre for Neurorehabilitation and Neuropsychiatry. We cover patients for the whole of Northeast, Cumbria up to Yorkshire, and we look after patients with neuropsychiatric conditions, which is, for example, Huntington's disease, Parkinson's disease, patients with acquired brain injury, head injury, hypoxic brain injury, autoimmune encephalitis, and management of these patients who have mental health problems and behavioral difficulties. We also have a neurorehabilitation as one of the core components, which is managed by a multidisciplinary team who are involved in rehabilitation of physical and cognitive needs of these patients, both as an inpatient and as an outpatient setting. So for the purposes of today's lecture, we will be looking at a couple of case scenarios from my experience, and then I was going to talk to you a little bit about autoimmune encephalitis and the importance of knowing about autoimmune encephalitis in psychiatry. If time permits, we can talk a little bit about the role of neuropsychiatry and neurorehabilitation in these patients. So moving on to the first case, this is a 69-year-old Cumbrian man who works in a caravan park as a maintenance manager and a tractor driver. He has no previous mental health issues. He was fit and well, no physical health issues either. A family noticed that there has been a change in his personality from May-June 2018. And he has become a bit more independent or clingy to his family members, fearful for his own safety and having panic attacks regularly, about 6 over a period of a few weeks. He had also expressed some paranoid ideation that he wasn't feeling safe and not wanting to leave his wife. He was worried about his safety. In addition to these issues, he also had issues with remembering certain events, and his personality, especially in terms of his temper, changed. He became short-tempered. So just after this, their family went on a pre-booked holiday where they were staying in a hotel he was with his wife, his daughter, and other extended family members. There he attempted to hit his wife, and just a few hours later he tried to strangle himself with a belt and throw himself off the 6th floor balcony. He was taken to the local hospital where they identified that he had cellulitis of his leg, and he was given diazepam, and then they returned home. Soon after returning here, he had been extremely paranoid, and he has been phoning his GP about 85 times in one day. He was worried that he was going to be harmed and someone was going to come and get him. This led to a psychiatric inpatient admission in July 2018, where he was started on olanzapine 5 mg BD with a diagnosis of a late onset psychosis. So in the psychiatric unit, there were some concerns about his whether this was a neurological origin, and an autoimmune encephalitis was suspected, and then he was transferred to an acute hospital for management of encephalitis. He was noticed to have tremor of his right upper limb and widespread fasciculation, and he had coarse right hand tremor as well. There were no other neurological signs. He had an MRI brain which was normal, did not show any inflammation of the medial temporal lobes. He had whole body CT, which is usually done in patients with encephalitis to rule out any secondary malignancy. He had a lung nodule of 5 mm in size. His bloods were sent for autoimmune antibodies, and he was noticed to have anti-Casper2 antibodies in the serum. We can talk a little bit more about the different types of antibodies when we talk about autoimmune encephalitis. He was initially started on prednisolone 40 mg daily, but there were no change in his presentation. So he was then given intravenous immunoglobulin, and there was some response, and he was a bit more settled. He was then transferred to the local community hospital, which is closer to his home. So he was staying in the community hospital until things settled. By September 2018, he was still in the community hospital. He attacked his wife on one occasion, and then he attempted to strangle a nurse on the ward. He was moved back to the medical unit, where he was felt to be disoriented and confused. He was given 2 further courses of intravenous immunoglobulin, and following this, he was referred to us for neurorehabilitation at Walkergate Park. So I saw him in November 2018 in the acute medical unit. He was in an old age ward along with other elderly patients. When we went to see him, his wife was there. He was very reluctant to leave his bay and come and see us. He was extremely anxious. As we talked a little bit more about how he was, he started to express paranoid ideation. He stated that he couldn't trust anybody, including the staff on the ward and us, and said that people could fake it. So he was actually paranoid over everybody. He was paranoid about his wife. The more we talked about these paranoid thoughts, he became much more agitated and difficult to settle down. So he was given oral lorazepam whilst we were there to settle his anxiety and agitation. So he was receiving input from the local rehabilitation services at that point in that hospital, but the purpose of transferring him to us was for management of the psychosis and for management of his cognition as well. Given that significant amount of paranoia that he had, which had not responded or responded to a limited extent only with intravenous immunoglobulin, we suggested that he is treated further in terms of his autoimmune encephalitis. So he received plasma exchange in January 2019, for which he had a very good response. He improved significantly There were no paranoid ideation that was noticed when I saw him again. There were ongoing cognitive difficulties and there were ongoing issues with anxiety, tremors, and anxiety from the family about what had happened and about whether they can have him back home or not. And because they have a larger family with younger children, so wife was quite concerned whether he could come back home. So there was still quite a big role for neurorehabilitation in terms of the assessment of his cognition, anxiety, and working with family in getting him back to his accommodation. So this is an example of a patient who presents with clear psychotic symptoms in someone who had autoimmune encephalitis. Towards the end, we will be able to look a little bit more about the neurorehabilitation and how we manage this patient on our ward. So let's move on to the second case, who I saw as an outpatient. So this is a 79-year-old lady who was working as a receptionist until then in a dental surgery. So initial presentation in March 2018 was a 5-week history of involuntary movements of the face and left arm. She noticed that her cognition was deteriorating gradually, or I should say the family noticed, and she had some unsteady episodes and falls. In March 2018, she was diagnosed as having LGI-1 encephalitis. Again, we'll come back to the different types of encephalitis in a bit. And she was treated with oral prednisolone, and the dose was tapered. She had a good response to oral prednisolone, and the dose was gradually reduced as an outpatient. So in her case, the initial ACE3 in March 2018 was 85, and then it gradually improved to 92 and 96 in September 2018. Her MRI showed periventricular ischemic changes and small vessel disease with the lacunar infarcts and local area of gliosis, which are all totally unrelated to the autoimmune encephalitis, would be my view. She had CT of the chest, abdomen, and pelvis, which was normal. Lumbar puncture was done, which was reasonably within normal limits. EEG was normal. There was no signs of encephalopathy or the typical delta wave brush that is seen in patients with encephalitis. So the extreme delta wave brush is when there is typical slow wave that is seen in patients with encephalitis. So she was referred to our services for management of her mental health following the treatment of encephalitis. She was initially seen by older people's services. She was referred by the neurologist to older people's services. She was diagnosed as having atypical depression and memory difficulties by their services. She also had facial dystonic episodes and commented to have anxiety and bizarre episodes. So I saw her in the outpatient clinic. She came with her husband. She was able to give a good history, reasonable history, but checking with her husband quite regularly about whether, for the time scales mainly. She was seen by the neurologist at that time, and she was previously recommended to take carbamazepine for a query diagnosis of an epilepsy following encephalitis. She was started on a very low dose of carbamazepine, about 12.5 milligrams daily. She did not take the carbamazepine because she stopped taking the carbamazepine after a very small dose, as she thought she developed side effects to it. So during the review, she was a bit tearful. She was able to stay throughout the review and engage in the review. Towards the end of the review, I discussed with them about what my views and what my impression was. I told her that my view was that it may be a depressive episode in addition to anxiety that she has, though the other differential diagnoses were whether it was epilepsy or is it a behavioral difficulty following brain injury. She was, when I was talking about diagnosis and medication, she described, or she experienced this episode, as they call it, where she becomes stiff, clinging on to her clothes and holding on to things, becoming tight, screwing her eyes up and not communicating for a few seconds. And then that ends with an incomprehensible sentence. And the husband confirmed that this is the typical sort of presentation that they see at home quite regularly, once or twice a day. And this happens sometimes in clusters, and then there'll be months when she's doing absolutely fine, going out with friends, going out with family, and she has no issues at all whatsoever. So after looking at this presentation, my impression was whether it was still an epilepsy or an absence seizure that was presenting this way, or whether this is an episode of anxiety. It is still unclear. The EEG was normal, which was done previously by the neurologist, so we didn't have to repeat the EEG at this point. So she had already been tried on carbamazepine, so I chose lamotrigine as the next choice, which would help with both the mood and the epilepsy. I started her on a very low dose, half the dose, with very slow titration, so she would not engage, and she said she developed side effects which I don't which are not the regular side effects that you would come across or expect in a patient with in a patient who takes lamotrigine. So her side effects may be something like the symptoms are worse, symptoms are bad, the episodes are more. So she didn't take lamotrigine. So then I referred her to the community neuropsychiatric nurses who see her frequently at home. They saw her and she got on well with one of the nurses. And that went on very well, and she remained symptom free for a period of time, for a few months, and she was going out with family and friends. So we were looking at whether we should be discharging her, and then there was another episode where she was not eating, not getting up from bed, and losing weight. And husband and family were really concerned. She was referred to the crisis team, because I think we are a tertiary service. We are a small team covering the whole of the Northeast, so we are not able to provide crisis services. So we referred her to the crisis team, psychiatric crisis team. So they saw her quite frequently at home, and hospital admission was considered because of carer stress. There were concerns about inconsistencies in her presentation, which settled, which was noticeable by the staff members. Of note was that when she was her response to discussion about inpatient admission, which changed her demeanor. So this episode, though we suggested venlafaxine, started her on a low dose of venlafaxine and gradually titrated with the support from the crisis team to reassure her that we don't usually see many side effects and she can take it without significant side effects. She settled quickly. She was on a low dose of venlafaxine, but then she responded. She came off the venlafaxine and responded without medication. She said she did not need our services anymore, and she can refer herself again if needed. So she was discharged from services. So this is a unique presentation of behavioral difficulties of someone who had an encephalitis, who was functioning very well before, who had an encephalitis, who is struggling to cope with changes in cognition and lifestyle. So having discussed a little bit about a couple of case scenarios, one of which we will come back to at the end, I think it's time to look at a little bit about autoimmune encephalitis. So let's look at a little bit in terms of what it involves, what it is, the types of antibodies that you come across, and what is the role of psychiatrists in knowing about autoimmune encephalitis and managing. So autoimmune encephalitis, widely known as limbic encephalitis, which I think is a misnomer. Limbic area is, as you know, is a wide area of the brain involving the medial temporal lobe, the striatum, the thalamus, hypothalamus, hippocampal formation, going up to the cingulate areas. So inflammation of the limbic area is a huge area, and it doesn't specify exactly where the inflammation is, whether it is in the basal ganglia or whether it is in the hippocampal region or medial temporal lobe, because I think every area has a specific role, and involvement of specific regions in the brain is going to have a different presentation, so it is important to know exactly where the inflammation is. So autoimmune encephalitis involves the body developing antibodies to the cell surface protein or the receptor or the ion channel for the antigens in the brain. So this could be either intracellular or extracellular. These patients develop neurological symptoms and can also develop psychiatric symptoms because of the areas involved. The limbic area, where the basal ganglia, hippocampus obviously involves the cognition, basal ganglia, a bit of cognition and motor movements, and then you have the thalamus, hypothalamus, which are all involved with the nucleus accumbens, and the other areas that are involved with the dopamine pathways, which can impact on psychosis and perceptual disturbances. So depending upon the area of involvement, they can have behavioral difficulties, they can have issues with their cognition, mood, etc. So anyone who is not aware of or not able to make sense of what is happening to them or around them in the world, they are going to present as someone who has behavioral difficulties because they can't make sense of what is happening. Why do we need to know about the psychiatric symptoms that can happen? Because psychiatric symptoms, we need to pick up the psychiatric symptoms, or patients who have autoimmune encephalitis and have psychiatric symptoms, to aid early diagnosis and treatment. There are a number of antibodies. You will be hearing about one antibody every day that's coming up new. The commonest ones, the first three that are in blue, are the ones that are commonly tested. The first one is N-methyl D-aspartate receptor, which is a receptor containing glycine and GABAergic components, which is one of the commonest antibody that you will come across. And then you have the LGI1, which is leucine-rich glioma inactivated protein. This is the one we talked about in the second, the lady. And the other one is the Casper2. These are the three common antibodies that you would come across. When you ask for a standard test for sending the blood for antibodies, these are the three antibodies that will be tested. The other antibodies, there are a number of antibodies. They may be tested if the patient is still symptomatic and the three antibodies in the serum are negative. The other antibodies are to glycine receptor, GABA A receptor, GABA B receptor, and the DPPX, which is dipeptidyl peptidase like protein. And then you have the AMPA receptor antibodies, and then you have dopamine receptor antibodies, which presents with movement disorder and psychosis in where it acts on the basal ganglia. And then you have voltage gated calcium channel and voltage gated potassium channel antibodies. The LGI1 and Caspar2 were previously called voltage gated potassium channel antibodies. That is they're not called it used to be a umbrella term for both those antibodies. Nowadays we don't use the term VGKC, but there is a separate antibody called voltage gated calcium channel antibody, and there are many more antibodies that we don't test. So the antibodies can be extracellular or intracellular. They could be either on the surface of the neurons, or they could be in the cell bodies. The ones that are on the extracellular are the extracellular cell surface proteins or the receptor channel proteins. What happens in these antibodies is that the antibodies attach to the antigen and alter the structure and function of these receptors, and thereby it interferes with their function and the cellular function, and that is how the damage happens. They have a better prognosis and it is reversible if it is identified early before significant damage is done to the neurons. In terms of intracellular, it is mostly paraneoplastic. About 75% of the cases they are paraneoplastic, and small cell carcinoma, lung carcinoma, ovarian and testicular carcinoma are the commonest ones. And mainly it's anti-WHO, anti-MA antibodies are the intracellular ones. Anti-WHO is more common than anti-MA, and anti-WHO is associated with small cell lung cancer. Anti-MA antibodies has a better prognosis. So because these antibodies enter the cell and then interact with the antigen, causing cellular damage, it causes significant neuronal cell loss and inflammation. Looking at this picture, it gives you an understanding of where the extracellular and intracellular antibodies work and what happens. So the blue pronged ones are the antibodies that attach to the red receptors on the cell surface antigen, thereby altering the cell function. The picture on the right shows the antibodies that react with the cytotoxic T cell that act on the cell and cause damage to the neuronal cell bodies and cause cell inflammation. So why do we need to diagnose? Why do we need to bother about this condition at all? It is a rare condition, but it's not that uncommon. About 5 to 8 per million cases happen is the incidence. 50% of the time an antibody is not identified, no specific antibody is identified. Because that is because we haven't identified an antibody that could be tested. Only in 50% of the cases you know the name of the antibody that is causing this inflammation. So this is autoimmune encephalitis is the third common cause of encephalitis, next to herpes simplex virus encephalitis and acute disseminated encephalomalacia. The most commonest antibody is the NMDA receptor antibody, which happens in about 1% of ITU admissions in young adults. This affects all age groups, especially young adults. So in terms of the psychiatric manifestations, 70% of the patients present with psychiatric manifestation. Anxiety, panic, behavioral disturbances, delusions, hallucinations, paranoia, psychosis. You could have any psychiatric disturbance with these presentations. the typical features are a prodromal flu-like symptom for a few days, a week before the illness, acute onset of psychiatric symptoms, and in patients with we all know that schizophrenia or first episode of psychosis usually happens in a younger male, early 20s or late teens. But what happens is in these patients, what we see is it could be a late presentation. It could be the age might not fit for example, as in the first patient, it may be that it happened in being presented at the age of 50. So it could be a change in behavior and any other neurological symptoms that you might come across, memory difficulties, altered level of consciousness, and autonomic disturbance. So in this table, it gives you an understanding of the different psychiatric presentations that these different antibodies can present with. NMDA receptor encephalitis mostly presents with psychiatric symptoms. 40% of the patients, the initial presentation is psychiatric symptoms, and in 80% of the cases, through the course of the illness, they will have some sort of psychiatric symptoms. So that tells you how important it is to be aware of these conditions. So LGI antibodies, the ones that are commonly seen, the LGI antibodies can present with behavioral changes, including apathy. Apathy is total lack of interest in the environment. So LGI patients can present with that as well. And Caspar-2 antibodies can present with sleep disturbances. I'm only talking about the unusual presentations that you might come across. In addition to that, they can have any other psychiatric symptom that can present in, as you see in your clinical practice. So, as we discussed before, the red flags, what would alert you to a diagnosis of autoimmune encephalitis in a patient with mental health problems? Sudden onset of any psychiatric presentation, anxiety, panic attack, depression, delusions or hallucinations. And what you will notice that there is no particular trigger. It's a bit unusual, and you don't, something's not fitting right. Maybe a prodrome of flu-like symptoms may be there. In terms of behavioral disturbances, change in behavior can be there. Cognitive deficits, memory difficulties, short-term memory problems may be present. Seizures, absent seizures, or generalized tonic-clonic seizures, fasciculations, tremors, myotonia can be there. Okay, so in terms of EEG, there can be extreme delta brush, which is typical of NMDAR encephalitis. It is seen in 90% of the patients with NMDAR encephalitis. Usually the difficulty that we may have is by the time we think that this is a neurological condition, I think patient may be extremely disturbed. We may not be able to send them for an EEG. But I think if any suspicion of any neurological condition is picked up by the psychiatric team, I would refer them to neurologist pretty soon rather than waiting for it to develop further. Okay. So in this table, it gives you a list of the psychiatric and neurological symptoms that was present in about 100 patients that they looked at. In this, what it shows is that behavioral changes are present in 60% of the patients, nearly 60% of the patients with encephalitis. And 70% of the patients have some sort of psychiatric disturbance. It could be any of the psychiatric symptoms, and then catatonia, and mutism, paranoia, anorexia was described as well. In terms of neurological symptoms, there's a wide range of neurological symptoms from memory difficulties, sensory motor disturbance, motor movement disorder, movement disturbances, and ataxia, diplopia, and 70% of the patients presented with neurological symptoms had neurological symptoms. So psychiatric symptoms are equally common in patients with autoimmune encephalitis during the course of their illness or as the initial presentation. So in looking a little bit more about different types of encephalitis, different antibodies. So NMDA receptor encephalitis antibody is to the cell surface antigen, which is highly expressed in the hippocampus, and hence the amnesic syndrome is associated with this condition. With the antibody acting on the receptor, it is internalized and it hypofunctions. There is a glycine and a GABA component. This is most common type of encephalitis that you would come across. It is associated with neoplasm in 60% of the patients. It involves the medial temporal lobe and corpus callosum. In the long term, it can lead to atrophy of the frontotemporal region. So LGI and Caspar-2 encephalitis. The NMDA receptor encephalitis starts as an acute presentation within a few days or weeks, but the LGI and Caspar encephalitis, they present as a gradual indolent course. LGI1 in patients present with hyponatremia in 65% of the cases, and the chance of underlying tumor is rare in these patients. They also develop what is called facio-brachial seizures, which is twitching and twitching of the face and hemiface and limb. And in this presence within the older age group, and the protein is identified in the hippocampal neuron. So in terms of Caspar-2 encephalitis, it presents with encephalopathy, autonomic symptoms, peripheral nerve excitability, where like our gentleman who had fasciculations, tremors seen in the which was ongoing for a while. So they have antibodies to the antigens in the juxtaparanodal region of the myelinated axons. Neuromyotonia may be associated with antibodies from the thymoma. Okay, so they have central and peripheral effects. Morvan syndrome have peripheral nerve excitability, dysautonomic symptoms, and neuromyotonia have firing of peripheral neurons. So both LGI and Caspar-2, they have an older age of onset and a gradual course in terms of the onset. So general features for most antibodies. The potential triggers may be being preceded by a viral encephalitis in 20% of the cases, and sometimes there may be associated tumors. 60% of these patients with any encephalitis have a low-grade fever, malaise, and headache. The prodrome can vary depending upon the type of the antibody. In terms of MRI, 30% of the cases show MRI changes, mostly seen in GABA B receptor antibodies. In terms of if this antibodies are negative in serum, then it is tested in the CSF, but not all antibodies are seen in the CSF. So antibodies to the glutamate N1 subunit is much more reliable in the CSF than to the serum. Most common CSF abnormality in LGI1 and DPPX, which is the dipeptidyl receptor antibodies. So the other encephalitis, AMPA receptor encephalitis, again late age of onset. GABA B receptor encephalitis, 50 to 60% have associated cancer. refractory epilepsy in GABA B receptor encephalitis, you may be very well aware that GABA is an inhibitory neurotransmitter in the brain. And if you have disturbance of the inhibitory neurotransmitter in the brain, then it is going to lead to increased excitability and seizures, which may be difficult to treat. Yeah, I think in DPPX antibodies, myoclonus, tremor, and exaggerated startle response is also present. So this table gives you a bit of an idea about what the MRI findings and how much it is positive, and the CSF abnormalities and whether it is detected in the CSF. So we'll look at the most common ones, the NMDA receptor encephalitis, where it is interesting to see that the brain MRI is normal in 50% of the patients, in more than 50% of the patients. And CSF shows lymphocytosis, and it may be negative as well. So in LGI1 encephalitis, the third down the list, the MRI changes may be abnormal in the medial temporal lobe in up to 60% of the patients, accentuated by flare. The CSF finding is not seen. CSF antibodies are not identified. Again, in Casper-2 encephalitis, the abnormality in medial temporal lobe in flare is seen in 40% of the patients, and the CSF abnormality, CSF antibodies are rarely identified. So this shows the importance of both the investigations and the clinical diagnosis to identify autoimmune encephalitis. This is not a scan from one of our patients, but this is how usually, if it is abnormal, this is what is seen. You can see the picture on the right, which is C. You can see the involvement and hyperintensity in the medial temporal lobes. So, having discussed a little bit about the autoimmune encephalitis as such, the clinical pathology and what happens, let's move on and talk a little bit about neurorehabilitation in patients with autoimmune encephalitis. So we provide both inpatient and outpatient rehabilitation in patients who have cognitive and physical difficulties, or just cognitive difficulties depending upon the level of impairment. Not all patients with autoimmune encephalitis need to be admitted for rehabilitation. Some of them do very well, and they can be managed with community services, which are available in certain areas and not in certain areas. Some patients start to recover, and the recovery is usually very good in the first 6 months, and then this continues for up to 2 years, and the neuroplasticity and helps in developing new circuits to be able to get back to their new baseline. So this is the garden inside the hospital for the patients to use. We have 4 inpatient wards. 2 are medical rehabilitation wards, and my ward is a rehabilitation ward with neurobehavioral difficulties. So we have patients with brain injury, autoimmune encephalitis, hypoxic brain injury, road traffic accidents, some complicated strokes, and then they come here for rehabilitation when they have either behavioral difficulties or additional mental health issues. So the long-term consequences of patients with autoimmune encephalitis is similar to patients who have TBI. They can have behavioral consequences, they can have cognitive difficulties, and they can have physical health difficulties. In terms of cognition, they can have an amnesic syndrome, they can have speech difficulties, executive dysfunction, they can have memory difficulties. That's the commonest one. And in terms of neuropsychiatric disturbances, they can have ongoing anxiety, mood disturbances, panic attacks, psychosis. Confabulation is something that usually presents as psychosis in patients with brain injury. They can also have issues related to epilepsy, and they may need neurorehabilitation prior to discharge home. In terms of long-term consequences, even the well-recovered, the best recovered patients may have some subtle cognitive difficulties. So the long-term, the neurorehabilitation is based on the model of neuroplasticity, that the patients will be, the brain will be able to make alternative circuits to be able to learn the skills that they may have been affected because of brain injury. So we provide outpatient or inpatient follow-up, as I told you before. So the rehabilitation will be either back to work or support with workplace, or going back to live with home, live with family, and providing the strategies and interventions that may be necessary to give the best for their lives. I'm probably going to skip this slide so that we can talk a little bit about the patient. So going back to the patient who we discussed, the first case 1, his initial, the gentleman who was Caravan Park's as a site manager, who had Casper-2 encephalitis, his initial ACE3 in July was 73, and then in August it dipped down to 64, and in September it went up to 84. So this, I would say, was affected by a few different things, probably because of the significant psychosis and also because of the cognitive difficulties that he had, which had recovered and which had improved later on. So neuropsychology assessment was done on the ward. He was admitted to our services for cognitive assessment and rehabilitation and discharged back to his home address. Premorbid IQ was average to low IQ. New learning difficulties mainly around verbal reasoning. And this improved in retest, which shows that he is learning, he is improving and his brain is recovering from the injury. He was low average on executive function difficulties, and verbal problem solving, he scored low average, and he needed help with novel and complex problems. So these are sort of like higher executive function difficulties. He was able to function on a day-to-day basis. He was able to look after himself. He did not need any support from nurses. He was able to choose his clothes. He was able to choose his what he wanted. But if he's left in a new environment, which is novel environment, then he might struggle. If he was asked to complete a task, then he might struggle. So it is those sort of difficulties that he might find it difficult. So in terms of neurorehabilitation, so we look at what the patient wants and what they want to achieve during this admission. So his aim was to go back home, manage his anxiety. So we worked on his cognitive rehabilitation. I stopped the olanzapine and lorazepam, which he was on for a long time because of the paranoia. He stayed well without it, and even the anxiety gradually improved when we started to engage him back with his family and support him in terms of discharge. He started to go home and meet his old friends, and he got on well, and as his confidence increased, his anxiety reduced. So I think we are coming towards the end of the presentation, and yeah, some of the references, there's plenty of literature around on autoimmune encephalitis, and you can always email me if you need to learn anything more about autoimmune encephalitis. Thank you very much for listening. I hope you have gained some useful information about autoimmune encephalitis, and I'll meet you for some questions.
Hello there. Right. Okay. Sorry, I had to close everything down there. Thank you for dedicating the time to make that make that for us, Gayatri. I know that we had a bit of fun trying to get that recorded. So we've had a few questions come through. And what we'll do now is we'll read those through in kind of like the chronological order. If anyone has any more questions, please do just put them in the chat. And I may have a question or one or two questions myself as well, if that's okay. So I suppose we'll start with the first one then. What percentage of patients with autoimmune encephalitis will die if they're not given the correct treatment?
Can you hear me, Reese?
I can, yeah.
Yeah, I haven't got the microphone, so I thought I'll try. First of all, I just had to apologize for the squeaky noises during the presentation. That wasn't my pet, that was my chair. So thanks for bearing with that. I noticed how evident it was when I was listening to the presentation today. So going back to the question about the mortality rate, that is what is the question, isn't it, from Laura?
Yes, that's right.
So if untreated, the mortality rate, it varies, but it says 6%. The earlier you treat, I think the better it is. As you can imagine, the injury to the brain is going to be less if it is picked up earlier. I don't personally treat patients in the acute phase, so I'm not particularly familiar with how much of my patients, what is the mortality rate in my patients. What I see is patients who have had encephalitis and who have come through the other side. If untreated, especially I think NMDA receptor encephalitis, they can go on to develop complications. I think status epilepticus can happen, and reduced conscious level can happen as well. So the mortality rate in one study was around 6%. So I think you may want to read a little bit more about it on that question if you want to know the exact mortality rate. But obviously, the earlier you pick up, you're going to treat it and the mortality is going to be less.
Mm-hmm. Okay. And another question here from Laura. Why is it that there are so few neuropsychiatrists, and what's the kind of typical pathway through which people end up seeing a neuropsychiatrist? How do they get referred?
I think I wouldn't say, yeah, I think there are every area, wherever you are within the UK, you should have a tertiary referral center for neuropsychiatrists. For accessing a neuropsychiatrist, and there is also, I think it's becoming much more standardized now that every major trauma center should have a neuroscience center, which should have access to neuropsychiatrists. We get referrals from everywhere. We get referrals from the GP, we get referrals from secondary care, we get referrals from neurologists, and secondary care psychiatrists can refer, any services can refer to us. And I don't think the psychiatrists will be offended if you bypass them and refer to the neuropsychiatrist, because many of our patients who come to us are referred directly. They are not referred by the psychiatrist. The referral from the psychiatrist forms about 10 to 20% of our referrals. Majority of them are from neurology or GP or other secondary care services.
Mm-hmm. Okay. This might be perhaps a good point just to ask, could you perhaps summarize the role of a neuropsychiatrist?
Okay. So what a neuropsychiatrist does is manage patients with mental health problems. We are basically trained in psychiatry. So our qualification is MRC Psych, but we have, like how you have old age psychiatry, adult psychiatry, neuropsychiatry is a branch of psychiatry, but we don't have an accreditation for it yet. There is, you don't get an accreditation. As liaison psychiatry, you would have an accreditation, but neuropsychiatry, you don't have an accreditation yet, but we are working on that. So, so what we do special interest, and we do an SPR training in neuropsychiatry, and then we start to work with the neuropsychiatry, and that is how the training pathway is. In terms of patients we see anybody with mental health problems, in a neurological condition or a neurodegenerative condition, or acquired brain injuries. a bit more specialized within neuropsychiatry is neurobehavioral neurorehabilitation, where neurorehabilitation for patients with acquired brain injury is needed for patients with mental health problems and behavioral problems. So that bit becomes a bit more psychiatry, but they still have a strong physical health rehab component to it. So neurobehavioral neurorehab is much more a complementary thing between the two, between physical health and mental health. So there are only a few centers with neurobehavioral neurorehabilitation.
Okay, thank you. I have a question here from Harry.
Mm-hmm.
If a patient presents with autoimmune encephalitis without a known history of cancer, would you go looking for possible tumours due to the high rates of association in some of the subtypes that you shared?
I think most patients that I have seen when they are in the acute hospital, they do have a whole body CT. If an autoimmune encephalitis is detected in both the patients that I have presented, they had a whole body CT. So they do a CT just in case and to rule out any hidden malignancy.
Mm-hmm. Okay. Harry also then went on to ask, also, is it known whether patients are more likely to develop those types of cancer in the future, even if they don't have them at the time of presentation?
I don't think that is something that people keep monitoring. No, it's at the time of diagnosis rather than whether they'll develop later, because the antibodies are sort of paraneoplastic thing. So it is from the tumor rather than them developing the tumor later on.
I see. Okay. And we spoke a little bit about kind of whole body CT, MRI. I wonder if a PET scan's used at all. I understand they're probably more on the research side, but is there any clinical use for them?
I think MRI shows acute changes. If you have later on, if you have any concern about the level of improvement and if they are not making progress, and if you feel that specific areas of the brain may be affected, or if there is a comorbid neurodegenerative dementing process going on, in those cases I might want to do a PET scan. Otherwise I don't usually do a PET scan.
Mm-hmm. Okay. Another question here from Laura. Is autoimmune encephalitis associated with thyroid autoantibodies at all?
The Casper2, the MOR1 syndrome with Casper2 encephalitis can have association with thymoma, but not particularly that thyroid antibodies are associated. But, if you have antibodies for one thing, you may, but that's not a common thing that we look for or find.
Mm-hmm. Okay, I think that's all of our questions. I'd just like to say a massive thank you again for developing that talk for us. And yeah, I speak on the behalf of Benjamin and Dee as well, in that, I understand that it was, as I say, we had some fun trying to get that recording done. Thank you to everybody else as well for tuning in this evening. I know it's been it's quite a late time. And as I think one of the questions at the beginning, we are hoping to kind of publish a recording of the night, but we can't say just when that will be. As soon as we have that sorted, we'll put it out onto our social media. Okay.
Thank you very much.
Thank you.
Take care. Have a good evening. Bye-bye.